All right, everybody. Let's look at neuromuscular blockers today. Before we get into this, I want to mention that this presentation is really going to be focused on the pathophysiology and physiology behind our neuromuscular blocking agents, the depolarizers and non-depolarizers. We're going to talk about reversal in depth later. However, we're going to touch on a few key points today, and to begin with, we're going to look at some commonly used neuromuscular terminology. So understanding this terminology, this is very useful when talking about neuromuscular monitoring, but also for administering these drugs. So these terms describe critical time points and ratios used to evaluate the onset, duration, and recovery from neuromuscular blockade. So onset time refers to the time from the administration until its maximal effect is observed. This is crucial for determining how quickly a muscle relaxant will take into effect. And this is important when you consider an emergency situation like an RSI or any critical situation. The clinical duration refers to the time it takes from the neuromuscular response to recover to 25% of its initial strength following neuromuscular blockade administration. We often assess the total duration of action, and this is a measure until you have a 90% recovery. Okay, so sometimes you'll see that 0.9, okay, so 90% recovery. Additionally, we have the recovery index, and this is the speed of recovery, specifically the time from 25 to 75% recovery of a muscle twitch response. And this gives you some insight into how quickly someone will regain muscle function. The most commonly used test that we're going to perform in anesthesia is the train of 4 ratio. Okay, and this is going to compare the strength of the fourth muscle twitch to the first. When the fourth twitch reaches 90% of the first, this is whenever you would have sufficient recovery. This is a key tool in monitoring for residual paralysis and ensuring you have a full recovery from neuromuscular blockade. All right, so keep these terms in mind as we move forward with the rest of this presentation. It is crucial that you understand the neuromuscular junction. This is a critical site where nerve impulses are transmitted from motor neurons to skeletal muscles, and this leads to muscle contraction. The transmission depends on the release of acetylcholine from synaptic vesicles. Synaptic vesicles right here in the presynaptic terminal, and these will bind to the nicotinic acetylcholine receptors in the postsynaptic muscle membrane. This interaction will trigger depolarization of the muscle membrane, and this leads to a cascade of events that all leads to muscle contraction. Understanding this neuromuscular junction is essential for you as an anesthesia provider because you're going to be giving these neuromuscular blocking agents. And these work by altering the acetylcholine activity at this junction. Building on that and maybe bringing back to memory some of the things we've covered in the past, acetylcholine is synthesized in the cytoplasm of the motor neuron and stored in vesicles until an action potential arrives. Upon arrival, voltage-gated calcium channels open. You now have calcium influx, and this will trigger acetylcholine release into the synaptic cleft. It will then bind to those nicotinic acetylcholine receptors on the muscle membrane, and this will initiate that contraction. After its action, it is rapidly hydrolyzed by acetylcholinesterase ASE, an enzyme. And this is to prevent continuous stimulation and allow for muscle relaxation. Neuromuscular blockers interfere with this process by either inhibiting acetylcholine receptors or by maintaining prolonged depolarization. So the neuromuscular junction is that synapse between a motor neuron and a muscle fiber. Acetylcholine is crucial for muscle contraction, and our neuromuscular blockers act on the acetylcholine receptors to block transmission. Muscles are broadly classified into 3 types: skeletal, cardiac, and smooth. Skeletal muscles are responsible for voluntary movements and are characterized by their striations, their striated appearance. They're controlled by the somatic nervous system and attached to bones via tendons, and this makes them essential for movement. These are innervated by motor neurons originating from the spinal cord or brainstem, and their primary function is converting chemical energy into a mechanical work during movement. You contrast that with smooth and cardiac muscles, which are controlled involuntarily by the autonomic, the automatic nervous system. Smooth muscles found in the walls of hollow organs like the GI tract and blood vessels. These facilitate processes like digestion and blood flow. Cardiac muscles are located exclusively in the heart and are responsible for pumping blood throughout the body. And while skeletal muscles contract quickly, smooth muscle contractions are a little slower and sustained. And cardiac muscle cells are synchronized by those intercalated discs. So with this in mind, when we're talking about our neuromuscular blockers, we're talking about skeletal muscle. Skeletal muscle is essential for voluntary movements and makes up around 40% of your body's mass. These are tubular and multinucleated, meaning that each muscle fiber has multiple nuclei. These muscles convert chemical energy, and they get this energy from ATP. They convert it into mechanical energy, facilitating movement. This process involves the contraction and relaxation of muscle fibers, which are innervated by motor neurons. That motor neuron will originate either in the spinal cord or brainstem and transmit signals throughout the neuromuscular junction to activate a muscle's contraction. These muscles play a critical role in varying physical activities, from basic movements like walking and lifting to something more complex like maintaining posture and balance. The skeletal muscle system is also involved in thermogenesis, generating heat as a byproduct of muscle activity, helping maintain homeostasis, our body's temperature. Motor neurons control these muscles via the somatic nervous system, and any disruption in this pathway can lead to neuromuscular disease affecting voluntary movement. So skeletal muscle is responsible for voluntary movements. It's about 40% of your body's mass, and it's innervated by motor neurons from the spinal cord or brainstem. Smooth muscle differs from skeletal muscle in a multitude of ways. It lacks striations, and this is why it has a smooth appearance under the microscope, and it's controlled involuntarily by the autonomic nervous system. Smooth muscle is found in the walls of hollow organs like the GI tract, blood vessels, the bladder, and its contractions are generally slower and sustained, enabling functions like moving food through the digestive tract or regulating blood vessel diameter. Smooth muscle contracts through a different mechanism than skeletal muscle. The contractions are more prolonged, and this is essential for maintaining the function of hollow organs over time without fatigue. This slow, sustained contraction is critical in peristalsis, which is going to move food through the intestines, or that regulation of blood flow by altering vessel diameter. Smooth muscle is non-striated and controlled by the autonomic nervous system. It's found in hollow organs like the GI tract and blood vessels, and the contractions are slower and sustained. Smooth muscle is classified into two different types. You have multi-unit and visceral, a single unit. Multi-unit smooth muscle is like that found in the iris of the eye and large arteries, and it's controlled by neural inputs and contracts in response to specific stimuli. Each smooth muscle fiber operates independently, and this allows for that precise control over small areas. Think about pupil constriction or the dilation. You contrast that with visceral smooth muscle, contracts as a single unit and is often found in the walls of the GI tract. This type of smooth muscle can generate spontaneous contractions, which are coordinated by electrical connections between cells through our gap junctions. And those spontaneous contractions, this is part of peristalsis. This is how food will move through the intestines in a coordinated manner. Calcium plays a very important role in the contraction of smooth muscle. The process begins when calcium ions enter into the smooth muscle cell from the extracellular space through those voltage-gated calcium channels. Once inside the cell, that calcium will then bind to calmodulin, and this will form a calcium-calmodulin complex. This complex activates myosin light chain kinase, MLCK. All right, and this phosphorylates the myosin heads, and this allows them to form cross bridges with actin and initiate contraction. This is distinct in contrast to the skeletal muscle contraction, where calcium interacts with troponin instead of calmodulin. The calcium-calmodulin complex in smooth muscle regulates the phosphorylation of myosin, making that contraction slower but more sustained in skeletal muscle. Now, after that contraction, the muscle relaxes when calcium is either pumped out of the cell or sequestered into the sarcoplasmic reticulum, allowing the muscle to return to its resting state. Now let's recap just for a second here. Calcium enters the smooth muscle cell and binds to calmodulin. This calcium-calmodulin complex activates that MLCK, and that was the myosin light chain kinase, which then phosphorylates. Okay, and phosphorylation, what that is, that's the addition of a phosphate group to a molecule. And this will change that function or the activity of it. Phosphorylation is oftentimes like an on and off switch and adjusts how it will interact with other molecules. So when the complex activates myosin light chain kinase, it will add a phosphate group to the myosin heads, and this changes their function and allows them to form that cross bridge with actin and initiate muscle contraction. And this is all part of calcium's role in smooth muscle. This is in contrast, okay? This is in contrast to skeletal muscle, because skeletal muscle will interact with troponin instead of calmodulin. The relaxation of smooth muscle occurs when calcium ions are removed from the cytoplasm of the muscle cell. Calcium is actively pumped either out of the cell or back into the sarcoplasmic reticulum. This decrease in intracellular calcium concentration reduces the activity of MLCK, or that myosin light chain kinase, and activates myosin light chain phosphatase, which will then dephosphorylate, remove that phosphate group from the myosin heads. Without phosphorylation, myosin cannot bind to actin, and this results in the cessation of cross-bridge cycling and muscle relaxation. So, the process of smooth muscle relaxation is slower than that of skeletal muscle due to that slower calcium reuptake and that longer duration of that cross-bridge detachment. This slower relaxation is characteristic of smooth muscle function. This allows for prolonged contractions that are essential for maintaining blood vessel tone, and think about peristalsis as well. So, smooth muscle relaxes when calcium is pumped out of the cell or into the sarcoplasmic reticulum. Okay, the myosin light chain phosphatase, it dephosphorylates myosin, okay, and that will stop that cross-bridge formation and cause relaxation. And keep in mind, smooth muscle relaxation, it's going to be slower than skeletal muscle, and that's going to give you those prolonged contractions. Denervation occurs when the nerve supply to a muscle is lost, and this will result in a rapid atrophy of skeletal muscle fibers. These fibers rely on a regular neural input to maintain their structure and function, and without it, the muscle will begin to degrade fairly quickly, and this will lead to muscle wasting and weakness. This process happens relatively quickly in skeletal muscle when you contrast that to smooth muscle. Smooth muscle, it can also be affected by denervation, atrophies slower, and become hypersensitive to neurotransmitters because of receptor upregulation. So muscle atrophy from denervation is typically accompanied by changes in muscle fiber composition. Muscle fibers switch from a fast twitch fiber, and these are for those rapid, powerful movements, to a slow twitch fiber. And these slow twitch fibers are more fatigue resistant. This impacts the muscle's functional capabilities, leading to reduced strength and endurance. And to add to that, protein degradation and changes in hormone synthesis contribute to the process of atrophy. This is why rehabilitation and reinnervation techniques are crucial in slowing down or potentially reversing atrophy. So denervation leads to rapid skeletal muscle atrophy due to a loss of neural input. Smooth muscle will atrophy slower, and it can become hypersensitive to our neurotransmitters. And muscle fiber composition changes during atrophy, and that will lead to a reduction in muscle strength. A motor unit consists of a single motor neuron and all the muscle fibers it innervates. The size of the motor unit varies depending on the type of movement it controls. So a small motor unit might be responsible for fine, more precise movements. Think about the eyes and the hands, while large motor units control powerful bulk movements. Think about your legs and your back. And each motor neuron branches out and connects with multiple muscle fibers, and this ensures coordinated muscle contraction. We talked about the two main types of muscle fibers, okay? We have that red slow twitch type 1 and that white fast twitch type 2 fibers. Those slow twitch fibers, they are fatigue resistant, They're rich in myoglobin and ideal for an endurance activity. Think about walking, maintaining your posture. You contrast that with that fast twitch muscle fiber, and this is designed for quick, forceful actions. Think about sprinting or lifting heavy objects. But those fast twitch type 2 fibers, they fatigue very rapidly. Okay? The composition of our motor units influences our overall function and endurance of that muscle group. Okay? So a motor unit is made up of one motor neuron and all of the muscle fibers it will innervate. Motor units can vary in size, and they control either fine movements or stronger bulk movements. Slow twitch fibers are suited for endurance, while fast twitch fibers are suited for quick, forceful actions. Now, the neuromuscular junction is composed of the presynaptic motor neuron, okay? Presynaptic motor neuron, that terminal, then the synaptic cleft, okay? Synaptic cleft right here, and then the postsynaptic muscle membrane. Okay? Communication across this will be initiated by an action potential as it travels down that motor neuron. And this will lead to the release of acetylcholine from those synaptic vesicles. It will then cross that synaptic cleft and bind to those nicotinic acetylcholine receptors right here on the postsynaptic membrane. This binding will cause depolarization and initiate an action potential that propagates across the muscle membrane, and this leads to muscle contraction. This unique architecture is part of an efficiency in the transmission of signals. The synaptic cleft, although tiny, plays a vital role by allowing rapid diffusion of neurotransmitters. Additionally, that postjunctional membrane is highly folded, and this increases the surface area, okay, for acetylcholine binding. Notice how it is here. Proper neuromuscular junction function is crucial for muscle movement, and any disruption can result in a neuromuscular disorder, and this is what we're targeting when we're talking about our neuromuscular blocking drugs, this postsynaptic receptors here. So the neuromuscular junction, you have your presynaptic terminal, your synaptic cleft, and then your postsynaptic membrane. Acetylcholine plays a key role in transmitting those signals across that neuromuscular junction. Okay, acetylcholine will be stored in those vesicles. All right, and then you have our neuromuscular blockers, and they're going to disrupt this process and induce paralysis. Okay, by specifically talking about those postsynaptic nicotinic acetylcholine receptors on that muscle membrane. Synaptic vesicles are small structures. We're zooming in now. We're going to focus on this. These are small structures within that presynaptic terminal of our motor neuron where that acetylcholine will be stored before it's released into the synaptic cleft. These are filled with somewhere between 5 and 10,000 molecules of acetylcholine, and they're synthesized in the cytoplasm of that neuron and are transported and packaged into those vesicles. Now, when that action potential will reach the presynaptic terminal, it will trigger voltage-gated calcium channels to open. Okay? And when those calcium channels open, calcium ions enter in. This influx is essential for the vesicle to fuse with the presynaptic membrane, a process facilitated by proteins like snares and release acetylcholine into the synaptic cleft. And when we talk about proteins like snares, what we're talking about are soluble attachment protein receptors. These are part of the fusion of vesicles to target membranes, and they help transport materials within and between cells. So once in the cleft, acetylcholine binds to those nicotinic receptors on the postsynaptic membrane, and they initiate that muscle contraction. After their release, synaptic vesicles are recycled through a process known as endocytosis. This process, involving proteins like clathrin and dynamin, what they do is the vesicle is now going to reform, and it will refill, repackage, recycle that acetylcholine, ready for that next action potential. This is all part of the vesicle cycle, and this is critical for maintaining consistent neuromuscular transmission, and this is targeted by our drugs affecting neurotransmitters when it comes to their release or recycling. Okay, so synaptic vesicles store and release acetylcholine upon stimulation by an action potential. Calcium influx is critical for that vesicle fusion with a presynaptic membrane, and these vesicles are recycled after that acetylcholine is released through a process called endocytosis. Now let's look at this neuromuscular junction a little closer. Think of the neuromuscular junction as a channel of communication between the nervous system and our muscle cells. The function is to allow the nervous system to control the contraction of muscles, and this represents an important structure in the regulation of many biological functions. Motor neurons are the neurons that directly control effector organs, muscle cells. When that motor neuron fires an action potential, so when the sum of the inputs exceeds a critical threshold, keep in mind that response, that action potential, it's all or nothing. Depolarization produces an electrical current, and it will travel in a unidirectional pathway down the length of the sarcolemma, and it will propagate to the muscle. Synapse, this is the point where the nerve and the muscle meet. That synaptic cleft is that small space between them. And the neuromuscular junction. So the neuromuscular junction, this is the combination of the prejunctional motor neuron, motor nerve ending, the synaptic cleft, and those highly folded post-junctional membranes. Okay? This is a region of connection between a motor neuron and a muscle cell. We have that narrow gap, that synaptic cleft, and our nerve endings are going to be foot-like. And the skeletal muscle membrane is highly folded in that post-junctional area. And here's just another picture. Looking at those snare complexes, the synaptic vesicle exocytosis endocytosis cycle. This is all part of a cycle, okay? And synaptic vesicles, when they're in that small membrane-bound sac within that presynaptic terminal, the motor neurons at the neuromuscular junction, they're holding that acetylcholine and waiting until that action potential will arrive. And when it arrives and reaches that terminal, voltage-gated calcium channels open, calcium ions rush into the neuron. That increase in intracellular calcium triggers a fusion of our synaptic vesicles with the presynaptic membrane, and these are mediated by those snare proteins that I mentioned earlier. All right, do you remember those soluble attachment protein receptors? So those snare proteins. And once it fuses, they will release, okay? They will release that stored acetylcholine, 5 to 10,000 molecules, into the synaptic cleft. This is exocytosis. It will then bind to those nicotinic acetylcholine receptors on the postsynaptic membrane, initiating a muscle contraction. After it's released from that membrane, the empty synaptic vesicles are recycled through endocytosis, where they're going to be refilled with some new neurotransmitters, some new acetylcholine, but also some recycled. And this is done for future nerve impulses. This is all critical for sustaining neuromuscular transmission during repetitive muscle contractions. So, what do our synaptic vesicles store? Acetylcholine. Where at? In the presynaptic terminal. What happens when calcium influx is triggered? It will trigger, calcium will rush in, and it will trigger vesicle fusion with the membrane. And when that vesicle fuses with the membrane, it's going to release acetylcholine into the synaptic cleft. These vesicles, they're recycled through endocytosis and exocytosis to maintain neuromuscular function and neurotransmitter release. The postsynaptic membrane at the neuromuscular junction contains those nicotinic acetylcholine receptors, and these are integral to the process of muscle contraction. Okay, these receptors have 5 subunits that form a central pore, and when acetylcholine binds to these receptors, they undergo a conformational change that will open that channel. This allows ions like sodium and calcium to flow through. This ion movement actually leads to depolarization of the muscle membrane and the initiation of an action potential. And once the muscle cell is depolarized, the action potential spreads across the sarcolemma and into the T-tubules, and this triggers the release of calcium from the sarcoplasmic reticulum. This calcium release is what ultimately enables muscle contraction. The rapid termination of acetylcholine's effect is crucial for controlled muscle movement. And this is achieved through acetylcholinesterase. I think ASE, an enzyme. Acetylcholinesterase breaks down acetylcholine in the synaptic cleft. Any abnormalities in our nicotinic acetylcholine receptor function or the breakdown of acetylcholine, those are foundational in neuromuscular diseases. Okay, and sometimes they are influenced by pharmacologic agents. Okay, so our nicotinic acetylcholine receptors are located at the postsynaptic membrane at the neuromuscular junction. Acetylcholine binding causes those receptors to open and ion movement occurs. This leads to depolarization. Acetylcholinesterase degrades acetylcholine to terminate that signal and allow for relaxation. The release of acetylcholine is highly regulated, and it's going to begin with that action potential arriving at the presynaptic terminal of the motor neuron. This action potential opens voltage-gated calcium channels, and calcium will enter into the neuron. This influx triggers the synaptic vesicles, which contain acetylcholine, to fuse with the presynaptic membrane, and this is facilitated by those snare proteins, that ensure that vesicle membrane merges with the presynaptic membrane, and this releases acetylcholine into the synaptic cleft. And once in the synaptic cleft, acetylcholine will quickly diffuse across and bind to our nicotinic acetylcholine receptors on the postsynaptic muscle membrane. And what happens is when it binds, this is going to open those ion channels, and this will lead to depolarization of the muscle membrane, and this will all trigger muscle contraction. Keep in mind, acetylcholine's action is short-lived. It's rapidly broken down by what? Acetylcholinesterase. And it's broken down to what? Acetate and choline. And this prevents prolonged stimulation and allows the muscle to relax. This is vital for normal muscle functioning. So, acetylcholine release is triggered by calcium influx after an action potential. We have those snare proteins that will facilitate vesicle fusion with the presynaptic membrane, and that's going to release acetylcholine into the synaptic cleft. Acetylcholine will be rapidly broken down by acetylcholinesterase to terminate muscle contraction. Now, let's look at this image. Acetylcholine is the neurotransmitter released by our motor neurons at the neuromuscular junction as part of triggering muscle contractions. It's stored in our synaptic vesicles within the presynaptic nerve terminal, and it's awaiting that action potential to arrive to stimulate its release. When it reaches the nerve terminal, our voltage-gated calcium channels open, and this will allow calcium to enter in and prompt fusion of the synaptic vesicles with the presynaptic membrane. The result of this is that dumping of acetylcholine into the synaptic cleft. It will then diffuse across the synaptic cleft and bind to the nicotinic acetylcholine receptors on the postsynaptic membrane of the muscle fiber. That binding of acetylcholine to our nicotinic acetylcholine receptors, this causes those receptors to open, and this allows for sodium ions to flow into the muscle fiber, leading to depolarization of the membrane, and once again that initiation of what? An action potential. This action potential will travel along the sarcolemma and trigger muscle contraction. After acetylcholine has completed its role in transmitting the signal, it's rapidly broken down by acetylcholinesterase. Acetylcholinesterase is an enzyme that's located in the synaptic cleft, and it will prevent continuous stimulation of our muscles. So, acetylcholine is released from our synaptic vesicles upon an action potential reaching the nerve terminal. Acetylcholine will bind to the nicotinic acetylcholine receptors, post-junctional, leading to muscle depolarization and contraction. Acetylcholinesterase. This will break down acetylcholine to terminate the signal and prevent prolonged contraction. Now, we have this process called excitation-contraction coupling. And this is where a nerve signal, the excitation, leads to muscle contraction. It starts with depolarization of the muscle membrane after acetylcholine will bind to our nicotinic acetylcholine receptors on the postsynaptic side of our neuromuscular junction. This depolarization will activate our voltage-gated sodium channels, propagating an action potential along the muscle's fiber membrane, the sarcolemma, and as it travels down the muscle fiber through our T-tubules, and this action potential will actually activate those voltage-sensitive dihydropyridine receptors located in the T-tubules. And this in turn triggers the release of calcium from the sarcoplasmic reticulum through ryanodine receptors. Remember those receptors, the ryanodine receptors. It's important for something later. The influx of calcium binds to troponin, and this causes a conformational change in the tropomyosin, exposing myosin binding sites on actin filaments. This allows cross-bridge cycling between actin and myosin. This leads to muscle contraction. This process is tightly regulated. And this is regulated so tightly because muscle contraction should only occur when appropriate, and it can be quickly terminated when calcium is resequestered into where? The sarcoplasmic reticulum. So, depolarization of the muscle membrane triggers action potentials in the muscle fiber. Calcium is released from where? The sarcoplasmic reticulum. Cross-bridge cycling between actin and myosin produces muscle contraction. Now, the postsynaptic receptors, the postsynaptic membrane at our neuromuscular junction contains those nicotinic acetylcholine receptors, and these are integral, to muscle contraction. These are pentameric structures. They have 5 subunits that have that central pore, and when acetylcholine will bind to these, they undergo that conformational change that will open that channel. This allows ions like sodium and calcium to flow through. This will lead to depolarization of the muscle membrane and the initiation of an action potential. Now, once that muscle cell is depolarized, the action potential spreads across the sarcolemma and into the T-tubules, and this will trigger the release of calcium from the sarcoplasmic reticulum. Now, when this calcium is released, this enables muscle contraction. Acetylcholine will be rapidly terminated, and this is essential for controlled muscle movement, and it's broken down by acetylcholinesterase. It breaks it down into the synaptic cleft. Any abnormalities of the nicotinic acetylcholine receptor functioning or the breakdown of acetylcholine results in neuromuscular diseases, or it can be influenced potentially by pharmacologic agents. So, nicotinic acetylcholine receptors are postsynaptic. Acetylcholine binding causes those pentameric, those 5, they're pentameric, so 5 subunits around that central pore. They open, and ion movement comes in, and this leads to depolarization. Acetylcholinesterase is an enzyme located in the synaptic cleft, and this will terminate the signal and allow for relaxation. Now let's look at these pentameric receptors a little closer. So our nicotinic acetylcholine receptors are made up of 5 subunits. We have 2 alpha subunits and a beta, delta, and epsilon, or potentially gamma in fetal muscle, but just really focus on those 2 alpha subunits. These receptors are ligand-gated ion channels. They open in response to the binding of acetylcholine to the binding sites located on the alpha subunits. The opening of the receptor allows for ions like sodium to flow into the muscle cell, leading to depolarization and muscle contraction. Now, in fetal muscle, the epsilon subunit is actually replaced by a gamma subunit. This alters the conductance properties of that channel. This changes how the receptor responds to neurotransmitters, impacting muscle excitability. The activation of these receptors is essential for normal muscle contraction and voluntary movement. Any dysfunction or alteration in our nicotinic acetylcholine receptors, such as maybe congenital myasthenic syndromes, can result in impaired muscle functioning and weakness. So our neuromuscular blockers specifically target our nicotinic acetylcholine receptors to induce that muscle paralysis during surgical procedures. So our pentameric nicotinic acetylcholine receptors have 5 subunits. The most important are going to be those 2 alpha. These are ligand-gated ion channels that allow ion flow, leading to muscle depolarization and contraction. And the fetal version has a gamma subunit, and this will affect its channel properties and the responsiveness it has to neurotransmitters. Now, our nicotinic acetylcholine receptors are sometimes termed adult, due to the expression contrasted with the fetal receptors. So now our adult receptors have 5 subunits, 2 alpha subunits, as well as a beta, a delta, and an epsilon. The fetal will have the gamma. Now, each postsynaptic adult motor site receives input from a single motor axon, and the adult nicotinic acetylcholine receptors are restricted in high concentration in the postsynaptic membrane. but they're virtually absent extrasynaptically. As with the fetal nicotinic acetylcholine receptors, the alpha subunit contains the recognition site for acetylcholine and all neuromuscular blocking agents. Notice here, okay, for this top-down perspective, our alpha subunits, okay, and our nicotinic, or on our nicotinic acetylcholine receptors, our alpha subunits, and where acetylcholine is binding. Now, our non-depolarizers, they work by our non-depolarizers work by competitively inhibiting the binding of acetylcholine to our nicotinic acetylcholine receptors at the neuromuscular junction. So agents like rocuronium and vecuronium, they bind to these acetylcholine receptors without activating it. This will prevent acetylcholine from binding and thereby inhibiting depolarization. This prevents muscle contraction, leading to muscle relaxation, and this is essential for a number of surgical procedures. The non-depolarizing agents are competitive antagonists. They compete for the binding site. They compete with acetylcholine for that alpha subunit. The blockade is reversible, and the effects of these agents can be terminated by administering acetylcholinesterase inhibitors, like neostigmine, which increases the amount of acetylcholine available to outcompete our neuromuscular blockers. These drugs are characterized by their slow onset, longer duration, and these are oftentimes what we're using for long-term paralysis, okay? So our non-depolarizers are going to be what we're going to use for long-term paralysis. You contrast that with our depolarizing agents, like succinylcholine, really just for short-term intubation. Okay, so non-depolarizing agents prevent acetylcholine from binding to those nicotinic acetylcholine receptors. They induce muscle relaxation without causing depolarization, and the blockade can be reversed with acetylcholinesterase inhibitors. Calcium plays a vital role in muscle contraction. When that action potential reaches the muscle fiber, it will trigger the release of calcium ions from the sarcoplasmic reticulum into the cytosol of the muscle cell. Those calcium ions bind to troponin, and troponin is going to be that regulatory protein that will control the position of tropomyosin on the actin filaments. Now, when calcium binds to troponin, it will cause a conformational change that shifts tropomyosin. This will expose the active sites on the actin filaments, and this is where our myosin heads can attach. Now, that interaction between actin and myosin forms on the basis of muscle contraction. So this is what we refer to as a cross-bridge cycle. Myosin heads bind to the exposed actin sites. They pull the actin filaments. They release ADP and phosphate, resulting in muscle contraction. Now, in order for relaxation to occur, calcium is actively transported back into the sarcoplasmic reticulum, reducing concentration within the cytosol. And this leads to the dissociation of calcium from troponin, allowing tropomyosin to cover that actin binding site, and this stops that cross-bridge cycling and relaxes the muscle. So calcium ions are released from the sarcoplasmic reticulum. They bind to troponin to initiate muscle contraction. Tropomyosin shifts, exposing binding sites on actin, and this allows for myosin to form those cross bridges. Muscle relaxation occurs when calcium is reabsorbed back into the sarcoplasmic reticulum. Now, let's talk about depolarization. The depolarization of the muscle membrane is initiated by the binding of acetylcholine to those nicotinic acetylcholine receptors on the postsynaptic membrane at the neuromuscular junction. This binding causes ion channels to open. Now, sodium will enter into the muscle fiber, leading to depolarization. This depolarization is critical as it generates an action potential that travels along the sarcolemma and through the T-tubules. This propagates the signal deep into the muscle fiber. As the action potential will travel along the muscle fiber, it triggers the release of calcium ions from the sarcoplasmic reticulum into the cytosol, and this is where calcium will bind to troponin, initiating that contraction cycle. Now, those voltage changes caused by depolarization are essential for opening the calcium channels on the sarcoplasmic reticulum, and any disruption in this process can prevent proper muscle contraction, leading to maybe weakness or paralysis. Now, once that contraction is complete, you have a reuptake of calcium into the sarcoplasmic reticulum, and this will lead to muscle relaxation. Okay, so acetylcholine binds to those nicotinic acetylcholine receptors. This will depolarize the muscle membrane and generate an action potential. This action potential will trigger calcium release from the sarcoplasmic reticulum, and this initiates contraction. Depolarization and calcium release are key for muscle contraction and relaxation. Excitation-contraction coupling is that physiological process that links the electrical excitation of the muscle fiber to that mechanical contraction of the muscle. This starts with the depolarization of the muscle membrane following the binding of acetylcholine to those nicotinic receptors. The depolarization spreads along the sarcolemma down the T-tubules, causing that sarcoplasmic reticulum to release calcium into the muscle cell's cytoplasm. Now, those calcium ions, they'll then bind to troponin, allowing actin and myosin to interact, and this initiates muscle contraction. The precision of that coordination of those electrical signals and calcium release ensures that our muscle contractions occur rapidly and in response to neural signals. Calcium acts as the key intermediary between the electrical signal and that mechanical response. Any disruption in this process, such as improper calcium release or reuptake, can impair muscle functioning. This can lead to fatigue or cramps. After contraction, calcium is pushed back, it's pumped back into the sarcoplasmic reticulum, and this allows our muscles to relax and reset for the next contraction. So excitation-contraction coupling will link the electrical excitation of the muscle to the mechanical contraction. Calcium is then released from the sarcoplasmic reticulum, and it binds to troponin, and this facilitates that actin-myosin interaction. And proper calcium regulation is crucial for a controlled muscle contraction. Acetylcholinesterase, this is located in the synaptic cleft of our neuromuscular junction, and this is part of terminating the action of acetylcholine. After acetylcholine is released into the synaptic cleft and binds to those receptors on the postsynaptic membrane to trigger that muscle contraction, acetylcholinesterase will rapidly hydrolyze acetylcholine into acetate and choline, so it's breaking it down. This degradation, this breakdown of acetylcholine prevents continuous stimulation of our muscles. This allows that neuromuscular junction to reset for subsequent signals. Now, that rapid breakdown of acetylcholine by acetylcholinesterase is essential for proper function. Without the action of acetylcholinesterase, acetylcholine would remain and stay in that synaptic cleft and just continue to stimulate muscle contraction. This would lead to prolonged depolarization and eventually muscle fatigue, then paralysis. Now, when you have a dysregulation of acetylcholinesterase, and this can be through genetic conditions or pharmacological inhibitors, this can lead to neuromuscular disorders like myasthenia gravis or maybe some nerve agents. So, acetylcholinesterase breaks down acetylcholine into acetate and choline, terminating its action. This occurs at the synaptic cleft. Acetylcholinesterase prevents continuous muscle contraction and allows for the neuromuscular junction to reset. And any dysregulation of acetylcholinesterase can lead to neuromuscular disorders and continuous muscle stimulation. Okay, now let's talk through some of these steps. Step 1, that action potential is propagated via a motor neuron to the axon terminal. The points of contact will be the motor neuron and the motor end plate. And we have the chemical synapse. There's no physical connection, but a transmission of an impulse by chemical interaction. Our voltage-gated calcium channels in the presynaptic nerve, they open up, and calcium ions will diffuse into the terminals. These calcium ions trigger the release of acetylcholine from the synaptic vesicles. Acetylcholine will then diffuse across the synaptic cleft of the motor end plate, and acetylcholine binds to those nicotinic acetylcholine receptors, and these are those ligand-gated ion channels. Okay, so acetylcholine is synthesized in that presynaptic nerve terminal from acetate and choline. Choline is taken up by the presynaptic neuron through the high-affinity choline transporters, while acetate is derived from acetyl coenzyme A, produced in the mitochondria. These two molecules combine to form acetylcholine through the action of the enzyme choline acetyltransferase. Now, once it's synthesized, it's stored in those synaptic vesicles within the presynaptic terminal. Okay, we have acetylcholine stored in two pools within the nerve terminal. There's the immediately available pool, and these are vesicles near the membrane ready for release, and then that reserve pool, okay? And those are vesicles further away that are mobilized as needed. Now, upon nerve stimulation, calcium ions enter the presynaptic terminal, promoting the release of acetylcholine from the immediately available pool in the synaptic cleft. Now, after release, it will then bind to those postsynaptic nicotinic receptors, initiate muscle contraction, and it will then be broken down in the synaptic cleft by acetylcholinesterase, and this allows that process to begin again to reset. So acetylcholine is synthesized from acetate and choline in the presynaptic terminal. It's stored in two pools. You have an immediately available pool, which are very close to releasing it, and then a reserve pool. And calcium influx triggers the release of acetylcholine into the synaptic cleft to bind with those nicotinic receptors. So, continuing on, we have those ligand-gated ion channels. They open. Sodium enters, potassium exits. You have an influx of sodium and outflow of potassium, and this causes that membrane potential to become more positive, okay, less negative. Now, that membrane potential reaches a threshold. An action potential, okay, that all or nothing response triggers along the sarcolemma, and guess what? The muscle fiber contracts. So after acetylcholine is released into the synaptic cleft, it binds to those nicotinic acetylcholine receptors on the postsynaptic membrane. It binds to both the alpha subunits, okay, causing a conformational change that will open the receptor's ion channel. Sodium ions rush into the muscle fiber, leading to depolarization of the muscle membrane. And as a result, an end plate potential is generated, and this can trigger an action potential if it hits that threshold. When a large amount of acetylcholine is released, the cumulative effect results in muscle membrane depolarization, and this leads to the excitation contraction process. Voltage-gated sodium channels on the muscle membrane propagate the action potential along the sarcolemma and into the T tubules, ultimately triggering the release of calcium ions from the sarcoplasmic reticulum. This release of calcium facilitates the interaction between actin and myosin filaments, and this all leads to muscle contraction. Okay, so acetylcholine will bind to those nicotinic receptors on the postsynaptic membrane. This allows that sodium influx and membrane depolarization. That depolarization results in an end plate potential, and this triggers an action potential. The action potential leads to calcium release and muscle contraction. All right, 47 minutes in, and now we're just talking about neuromuscular blockers. I told you this would be a fairly lengthy presentation. I warned you ahead of time. But this is going to be so beneficial. This is a foundational piece of your anesthetic knowledge. I want this to be so solid for you all. I want you to really build on this. So when you get into the clinical setting, not only do you recognize this, but you understand it and you can talk through these settings. So our neuromuscular blockers, they are used to facilitate muscle relaxation. This aids in tracheal intubation, optimizes mechanical ventilation, and these allow surgeons to work more efficiently without the interference of involuntary muscle movements. During intubation, this relaxes and opens the vocal cords. This makes it easier and less traumatic. Now, the choice of a neuromuscular blocker depends on factors like the duration of surgery, the patient's condition, what side effects they have. Our depolarizing agent, succinylcholine, is often used for a rapid sequence induction due to that quick onset and short duration. You contrast that with our non-depolarizers like rocuronium and vecuronium, which are used for longer surgeries due to their prolonged effects. And you have to monitor these drugs to ensure that we are dosing appropriately and avoid complications like residual paralysis after surgery. So why do we use neuromuscular blockers? They make life easier, okay? Neuromuscular blockers, they enable surgeons to work better. They make intubation easier. They make ventilation easier. But you have to keep in mind the agent you're using, its onset time, its duration, and its side effects. Now, these are essential agents in anesthesia. These work by acting on those nicotinic acetylcholine receptors at the neuromuscular junction. And there are two main types of neuromuscular blockers. We have depolarizing, which is succinylcholine, all right, and then non-depolarizing, which is everything else. Depolarizing agents like succinylcholine mimic the action of acetylcholine by binding to those nicotinic acetylcholine receptors, causing prolonged depolarization. This results in an initial contraction, a fasciculation, followed by paralysis, as the muscle cannot repolarize properly. Now you contrast that with your non-depolarizers, like rocuronium, vecuronium, pancuronium, and they work by competing with acetylcholine for binding at that nicotinic acetylcholinergic receptor site. And when it binds to it, it doesn't activate it. When it occupies this receptor, non-depolarizing agents inhibit acetylcholine from triggering muscle contraction. This leads to relaxation and paralysis. These drugs are widely used, commonly used to facilitate intubation, ensure immobility during surgeries, and optimize operating conditions. So, non-depolarizers, okay? Depolarizers. Let's recap. So, both of these work on the nicotinic acetylcholinergic receptor at the neuromuscular junction. Depolarizing agents, succinylcholine, cause prolonged activation. Okay, you get a fasciculation, then you get paralysis. Non-depolarizing agents, rocuronium, pancuronium, vecuronium, everything else, competes with acetylcholine to inhibit muscle contraction. So our non-depolarizers, roc, vec, pancuronium, okay, they work by competing with acetylcholine for binding to those nicotinic acetylcholine receptors at the neuromuscular junction. They do not activate the receptor when it's bound. This prevents depolarization of the muscle membrane, and this blocks muscle contraction. It doesn't occur. The clinical effects include muscle paralysis, and this is essential for surgical procedures that require immobility and controlled muscle relaxation for a period of time. Pretty much anything in laparoscopy, you're going to relax. The onset, duration, metabolism of these drugs can vary, and you may use one over another. Rocuronium has a faster onset and an immediate duration, which makes it ideal for a rapid sequence intubation where you cannot use succinylcholine. You contrast that with, say, pancuronium. Okay, it's not one you see very often. I've never even given pancuronium. Okay, it's a longer acting drug. Okay, it's useful for procedures that require prolonged relaxation, but it has some side effects. Okay, it has some tachycardia side effects due to the vagolytic properties and the sympathomimetic properties of it. Okay, you have to keep in mind the effect of these agents. Okay, they can be reversed with acetylcholinesterase inhibitors like neostigmine or sugamidex, which will actually bind to that complex. Okay, and this leads to an increase in acetylcholine at the neuromuscular junction, and this outcompetes the neuromuscular blocker. Non-depolarizers compete with acetylcholine but do not cause depolarization. Non-depolarizer, not causing depolarization. These are used for surgery, intubation, and ventilation. Rocuronium is going to be quicker acting, pancuronium is slower acting. Non-depolarizing neuromuscular blockers prevent muscle contraction because when it's bound at that nicotinic acetylcholine receptor, it doesn't activate it. Okay, these are competitive antagonists. They compete with acetylcholine for that same binding site. Since they're not causing depolarization, there's not an ion channel that's opening. There's no influx of sodium ions. There's no subsequent muscle contraction. By blocking acetylcholine, we effectively induce muscle relaxation and paralysis. In procedures where prolonged paralysis is needed, non-depolarizers are the choice agents. The reversal of their effects can be achieved by using acetylcholinesterase inhibitors, like neostigmine, or true reversal agents like sugamidex for rocuronium. And both of those will lead to an increase in acetylcholine at the neuromuscular junction, outcompeting the neuromuscular blocker for receptor binding. So, non-depolarizers work by competitive antagonism. They're competitive antagonists. They bind to those receptors and don't activate them. This prevents acetylcholine from inducing muscle contraction, leads to paralysis. Reversal can be achieved with acetylcholinesterase inhibitors. Now, the effects of neuromuscular blockade. This is paralysis, and it begins with our small, rapidly moving muscles, so those are the eyes of the face. Then this progresses to our larger muscles, responsible for limb movement, and then finally the diaphragm. Now, this ordered progression is essential to monitor for the level of muscle relaxation during surgery. Neuromuscular blocking agents are used to facilitate intubation, maintain immobility, optimize mechanical ventilation, and sometimes we're even using it in post-op in the ICU. Now, recovery from blockade occurs in the reverse order, with the diaphragm actually recovering first. Then you have limb muscles, and finally smaller facial muscles. You have to carefully monitor this recovery process to ensure that patients are able to breathe independently before we extubate them. And we use techniques like the train of 4 simulation to assess the degree of blockade and ensure safe recovery and ensure that we are actively monitoring our patients, because this helps prevent complications from residual neuromuscular blockade. And this can lead to respiratory failure postoperatively, postoperative pneumonia, nausea and vomiting. Patients don't feel well if they're still a little weak, okay? That's why it's so important to monitor that. And we're going to get into monitoring more later, but just keep that in mind. Now, the pharmacokinetics of our neuromuscular blockers, these vary depending on the agent we're using. Succinylcholine has a very rapid onset of action and a very short duration, most of the time, making it ideal for a rapid sequence intubation. However, their shorter duration, okay, is due to that metabolism by plasma cholinesterase, because it quickly hydrolyzes the drug. You contrast that with our non-depolarizers, like ROC, a little slower onset, but a longer duration of action. And these are metabolized by either the liver or kidneys, depending on the specific drug. So take rocuronium, for example. It's going to be eliminated through hepatic metabolism, while vecuronium and pancuronium are cleared by the liver and kidneys. The duration of action of these agents can be prolonged in patients with hepatic or renal dysfunction, so you sometimes have to make dosage adjustments. So other factors like hypothermia, use of anesthetic agents, these can influence the metabolism and the duration of our neuromuscular blockers, complicating their pharmacokinetics. So succinylcholine, rapid onset, short duration of action due to plasma cholinesterase metabolism, pseudocholinesterase. Now, rocuronium has a longer duration, and it's metabolized by the liver. Hepatic and renal dysfunction can prolong the action of our non-depolarizers. All of our neuromuscular blockers are quaternary ammonium compounds, and this makes them water-soluble and limits their ability to cross lipid membranes like the blood-brain barrier. This is crucial as it ensures that these drugs act peripherally on muscles rather than the CNS. These show an inverse relationship between their potency and onset of action. So highly potent drugs have a slower onset, compared to lower potency drugs have a faster onset. Non-depolarizing muscle relaxants work by blocking acetylcholine from binding to the nicotinic acetylcholine receptor at the neuromuscular junction. And it just has to bind to one of the receptor's alpha subunit to block that channel and prevent ion flow, inhibiting muscle contraction. So rocuronium, vecuronium, pancuronium, atracurium, cisatracurium, they all have a varying onset and duration. They all have some side effects you have to keep in mind, and it makes them suitable for some clinical scenarios, some situations. Okay, so all neuromuscular blockers are quaternary ammonium, means they're water soluble. There's an inverse relationship between drug potency and onset, and non-depolarizers block the receptor by binding to just one single subunit, preventing muscle contraction. Now, neuromuscular monitoring is essential. We've talked about it a little bit, but this is employed through a nerve stimulator, and we have a number of techniques. Okay, the train of 4 is going to be the most common. This is going to be those 4 electrical impulses to a motor nerve, and it's a measurement of a corresponding muscle response. And we're really comparing the ratio of the 4th twitch to the 1st twitch to determine the extent of neuromuscular blockade. And this will help you to adjust the dosage of your drugs to ensure the patient maintains adequate muscle relaxation without any residual paralysis. Proper monitoring is crucial to avoid underdosing, which can cause patient movement. That's never a fun time in the OR when the surgeon, they're awake. They're not awake, they're just moving. Movement's a sign of life. Okay? Or potentially overdose, which can lead someone weak, which can lead someone to have some post-operative complications, some prolonged recovery. You have to monitor this blockade, and this way we can titrate our muscle relaxants, administer our reversal agents at the appropriate time to ensure we have a smooth and safe emergence from anesthesia. You don't want to say speed is crucial, but truthfully, one of the ways that you can truly assess whether someone is a good anesthetist is if they can have a replicatable, all right, smooth and efficient emergence the majority of the time. How quick you can go from one case to another is oftentimes what a good anesthetist is judged on. So non-depolarizers are often used for prolonged relaxation during surgery. Rockvec, cisatracurium. These drugs work by competitively inhibiting acetylcholine at the neuromuscular junction, preventing contraction. Okay? Where prolonged muscle relaxation is needed, this is where you're going to use a non-depolarizer. If it's just for intubation, succinylcholine is sometimes used. Now, the effects of these agents can be reversed with neostigmine, or the more specific reversal agents, Sagamedex. And Sagamedex is made for our steroidal, and we're going to get into that too a little bit, our steroidal compounds. All of our muscle relaxants, okay, are favored for their predictability and controllability in the operating room. Now, there are scenarios when we're going to use one over the other, but they are fairly predictable. Their use is widespread today, okay? You're going to be using paralytics. You're going to be using neuromuscular blocking agents. And we adjust our dosages based off of our monitoring, based off of the type of reversal agent we have, based off of the procedure we're doing. And we want to make sure that we minimize postoperative complications like residual neuromuscular blockade. Now, contrast that to succinylcholine. This is the only depolarizing neuromuscular blocker currently in clinical use. It works by mimicking acetylcholine and binding to that nicotinic acetylcholine receptor. And this causes continuous depolarization of the muscle membrane. This prolonged depolarization leads to muscle paralysis because the muscle membrane cannot repolarize and it cannot generate further action potentials. Succinylcholine is great for a rapid sequence intubation because it has a fast onset and a short duration. However, big however here, succinylcholine has several risks. Hyperkalemia. Succinylcholine is a malignant hyperthermia trigger. It can cause fasciculations, which cause postoperative myalgias. You have to be careful in patients with burns, neuromuscular diseases, renal failure. These are patients who can be at a high risk of developing hyperkalemia after succinylcholine administration due to that upregulation of those extrajunctional acetylcholinergic receptors. While the rapid onset makes succinylcholine ideal in certain situations, its side effects and contraindications limit its use in many patient populations. Okay? You want a Dr. Harmon opinion? Sucks. Okay? I'm not a fan of it. Okay? I only use it whenever I have to. All right, and to recap, it's a depolarizer, okay? It causes continuous depolarization, leading to paralysis. We like it for rapid sequence intubations or whenever you need to do nerve monitoring afterwards. You want that fast onset, short duration. But it has risks: hyperkalemia, MH, postoperative myalgias, to just name a few. Depolarizing neuromuscular blockers, so succinylcholine, mimics acetylcholine, and it binds to the nicotinic acetylcholine receptors at the neuromuscular junction. Succinylcholine causes continuous depolarization of that muscle membrane. This prolonged depolarization prevents the muscle from repolarizing. This leads to a phase of initial muscle contraction followed by sustained paralysis. And this is why it's so nice for a rapid sequence intubation, because it has such a quick onset. Now, the mechanism of action of these drugs can cause side effects, fasciculations. These are brief involuntary muscle contractions before paralysis sets in. Those lead to myalgias. Additionally, succinylcholine is oftentimes associated with hyperkalemia. You have a disruption in your potassium. Now, normally it's a small shift, but in certain situations it can be a large shift. It can be life-threatening. It's also a malignant hyperthermia trigger. So, even though it's rapid onset and short duration of action, is normally how it works. Sometimes succinylcholine is not the ideal drug. Sometimes you don't know that until you've given it. And we'll talk about that. We'll learn about that here in just a little bit. Okay, so recap. Succinylcholine is our only depolarizer that we're using today. It causes continuous depolarization, leads to paralysis. Quick onset, we love it for a rapid sequence intubation. Can cause fasciculations, can cause hyperkalemia, can trigger MH. Now, succinylcholine, it can produce prolonged depolarization at the end plate, and this can result in desensitization of nicotinic acetylcholine receptors, inactivation of our voltage-gated sodium channels at our neuromuscular junction, and this can increase potassium permeability in the surrounding membrane. The end result is failure of an action potential to generate due to membrane hyperpolarization and muscle blockade. Okay? These are acetylcholine receptor agonist. So succinylcholine, depolarizing muscle relaxant, acts as acetylcholine receptor agonist by binding to that acetylcholine receptor, that nicotinic acetylcholine receptor, at the motor end plate and generating an action potential. However, they are resistant to and they're not metabolized by acetylcholinesterase. This leads to a persistent depolarization of the muscle fibers, and this can result in a well-recognized fasciculation and paralysis. After it binds to that motor end plate receptor, it remains bound, and thus the end plate cannot repolarize. This is known as a phase 1 block. It is during this depolarizing phase that transient muscle fasciculation occurs. After adequate depolarization, you can have a phase 2, a desensitizing phase. And this is when the muscles are no longer receptive to acetylcholine released by the motor neurons. It's at this point that the depolarizing agent has fully achieved paralysis. The acetylcholine receptors can also undergo conformational and ionic changes after a period of time, and this results in a phase 2 block. Okay, that's different. So, depolarizing muscle relaxants act as acetylcholine receptor agonists. They trigger muscle contraction, and this renders that acetylcholine receptor inactive. They closely resemble acetylcholine. Succinylcholine is actually 2 acetylcholine molecules bound together. That's literally what it looks like. It's what it's chemically made up of. And it generates a muscle action potential. Now, succinylcholine molecules will diffuse away from the neuromuscular junction, and this can limit the duration of action. Remember, depolarizing muscle relaxants are not metabolized by acetylcholinesterase. So they will eventually diffuse away from the neuromuscular junction, and they are hydrolyzed in the plasma and the liver by another enzyme, pseudocholinesterase. Pseudocholinesterase. Remember that. So, clinical uses of SUCs. Rapid sequence induction. Emergency settings, boom, quick onset, 30 seconds. Short duration of action, most of the time. It's ideal for when you need to secure the airway quickly, or you have a case where you do not need muscle relaxation after intubation. So patients who are at risk of aspiration, who need immediate airway control, this can be great because it rapidly depolarizes that motor end plate, and this can cause a brief period of muscle fasciculation followed by flaccid paralysis. It can cause side effects, the hyperkalemia. This can be life-threatening, especially in patients with burns, neuromuscular disorders, or trauma. It can also trigger malignant hyperthermia in susceptible individuals. And we're going to talk about that here in a little bit. For these reasons, succinylcholine should be very carefully used. And maybe an alternative non-depolarizer, maybe rocuronium, might be preferred in certain patients. I RSI a lot of people with rock. It works just fine. So succinylcholine is often used for a rapid sequence intubation or induction. It causes brief fasciculations followed by paralysis, and it can cause some serious side effects: hyperkalemia, malignant hyperthermia, myalgias. It's not a clean drug. Succinylcholine, it's our depolarizer. It mimics acetylcholine at the neuromuscular junction, and its molecular structure is closely related to that of acetylcholine. The key difference is the longer chain allows for prolonged depolarization and blockade of that end plate. The quaternary ammonium group in succinylcholine is responsible for that strong positive charge, and this is crucial in binding to that nicotinic acetylcholine receptor. The metabolism of it is dependent on pseudocholinesterase, and that's going to be the enzyme that hydrolyzes succinylcholine. However, big however here, and we're going to build on this, you can have people with pseudocholinesterase deficiency, and this can lead to prolonged paralysis, posing significant risks. This is why a dose range of 1 to 1.5 milligram per kilo is recommended for intubation, because ideally the drug will have a rapid onset, short duration of action under normal metabolic conditions. Okay, so succinylcholine, it's a quaternary ammonium, strong positive charge. Acetylcholine and sucs have similar structures but differ in their duration of action and their metabolic pathways. Succinylcholine binds to that nicotinic acetylcholine receptor. It mimics acetylcholine and causes sustained depolarization with a fasciculation and then paralysis. It is ideally rapidly metabolized by acetylcholinesterase, but you can have patients who have enzymatic deficiencies. And the normal RSI dose is 1 to 1.5 milligram per kilo. Succinylcholine undergoes ester hydrolysis by pseudocholinesterase. And ester hydrolysis is a reaction that breaks an ester bond with a molecule of water. And the reason we have that short duration of action is the rapid hydrolysis by pseudocholinesterase or butyrcholinesterase or plasma cholinesterase. These are all similar names for the same thing. It's broken down into succinyl monocholine and choline. And this is interesting because only about 10% of the administered drug actually reaches the neuromuscular junction. Succinyl monocholine is a much weaker neuromuscular blocking agent, and it's metabolized a lot slower to succinic acid and choline. So keep in mind, plasmacholinesterase, butyrcholinesterase, pseudocholinesterase, all the same enzyme. Okay? These are enzymes that are going to break molecules down. So when we say succinylcholine undergoes ester hydrolysis by pseudocholinesterase, you should know that succinylcholine is broken down by breaking an ester bond with a molecule of water. Okay? And what's it broken down to? It's broken down to, I hope you answered, succinyl monocholine and choline. So keep in mind, succinylcholine will mimic the action of acetylcholine at the neuromuscular junction by binding to that nicotinic acetylcholine receptor on the motor implant. It's going to bind to both. This is going to lead to prolonged depolarization. This will prevent further action potentials. This rapid onset and short duration makes it highly useful in some situations. The metabolism of it is unique because it's hydrolyzed by pseudocholinesterase, also called butyrcholinesterase and plasmacholinesterase. That is not acetylcholinesterase, though. Okay? This distinction is critical because pseudocholinesterase deficiency occurs. This can be congenital or acquired and results in prolonged paralysis due to the inability to break this drug down. Therefore, you have to be very careful with who you use succinylcholine on. It's one reason why I personally like to avoid it at all costs, because you don't always know who has pseudocholinesterase deficiency. Now, one other key point here that I want to emphasize. Notice how I have that ED95. Think back, the effective dose, ED95. It's actually only 0.3 milligrams per kilo. Okay? So why do we use 1 to 1.5? Because when you give such a large dose, you get that rapid paralysis. Okay? So in patients with pseudocholinesterase deficiency, you can have a prolonged effect. Okay? But when you're bolusing 1 to 1.5 milligram per kilo, you're giving multiple times the ED95, so you're going to get that quick paralysis. And there's this picture of acetylcholine and succinylcholine. So acetylcholine is naturally occurring, that's going to bind to that nicotinic receptor. It's going to lead to muscle contraction. Succinylcholine mimics this, okay? And it mimics it because it's literally just two acetylcholine molecules fused together. This allows it to bind to those same receptors. But instead of being rapidly broken down, it remains attached and stuck there. This results in prolonged depolarization. This extended interaction leads to sustained paralysis of our muscle fibers. However, it's not stuck there for long as someone has pseudocholinesterase. Not acetylcholinesterase, pseudocholinesterase. Okay? The larger structure of succinylcholine is why it is resistant to rapid breakdown at the neuromuscular junction. Okay? This is why succinylcholine is a short-term muscle relaxant. It's useful for rapid onset and offset if a short duration of muscle relaxation is desired, like, okay, intubation, or rapid sequence induction. Okay, so recap. Succinylcholine is two acetylcholine molecules linked together. Prolonged action, okay? Unlike acetylcholine, which is quickly degraded by acetylcholinesterase, succinylcholine is broken down more slowly by pseudocholinesterase, leading to that sustained depolarizing effect. Okay? This is why it's normally fast-acting, but you can have pseudocholinesterase deficiency. Succinylcholine requires only 25% occupancy, okay, versus our non-depolarizers about 75%. Now, our depolarizing neuromuscular blockers, like SUCs, you get prolonged depolarization of the end plate. That's going to result in desensitization of those nicotinic acetylcholine receptors, inactivation of our voltage-gated sodium channels, and an increase in potassium permeability in the surrounding membranes. The end result is a failure of action potential generation due to that membrane hyperpolarization. So, with respect to our neuromuscular pharmacology, the two enzymes of importance that you have to know, they both are known to hydrolyze cholinesters. That's acetylcholinesterase and butyrylcholinesterase, or pseudocholinesterase. Acetylcholinesterase, okay? This is sometimes known as true cholinesterase, and this is very similar to our red blood cell cholinesterase. Acetylcholinesterase is present in the neuromuscular junction and responsible for the rapid hydrolysis of acetylcholine into acetic acid and choline. Butyrylcholinesterase, pseudocholinesterase, or plasma cholinesterase is synthesized in the liver. It catalyzes the hydrolysis of succinylcholine, which is going to occur mainly in the plasma after that molecule releases itself. So this is why it's a very unique neuromuscular blocking agent. It has a rapid onset of action. This is typically about 30 seconds to 1 minute. And this is its agonist effect on the nicotinic acetylcholine receptors. Since it mimics acetylcholine, only a small percentage of that administered dose even reaches the neuromuscular junction, about 10%. But it acts very quickly on that receptor site. Succinylcholine is metabolized by pseudocholinesterase with a plasma half-life somewhere around 2 to 4 minutes, so that clinical duration of 5 to 10 minutes is about what you see. And this ensures rapid metabolism. However, you can have patients who have pseudocholinesterase deficiency or dysfunction, and they will experience a prolonged paralysis because of succinylcholine. It cannot be broken down in these patients. It takes so much longer. It can last significantly longer, hours, sometimes hours and hours. This is essential to screen for conditions that affect cholinesterase activity before administration. Additionally, the drug's action can be affected by temperature, with cold environments slowing its breakdown, and there are some patients that you need to avoid it on due to the potential serious risks. Okay? So, succinylcholine, only 10% of it is actually going to reach the neuromuscular junction. It has a rapid onset and offset. The ED95, okay, is only about 0.3 milligram per kilo, but our RSI dose is 1 to 1.5. The plasma half-life is 2 to 4 minutes. Clinical duration of action maybe 5 to 10. And pseudocholinesterase breaks it down. So now, contrast this just a little bit. Our nicotinic receptors are a type of cholinergic receptors that respond to acetylcholine. These are ionotropic, and they, once activated, open an ion channel, and this leads to depolarization of the postsynaptic membrane. There are two main types of nicotinic receptors, and they're NM, okay, or NN, okay? NM receptors are found primarily at the neuromuscular junction, neuromuscular receptors, where they mediate contraction of skeletal muscle, okay? NN, okay, N2 receptors are located in the autonomic ganglia, the adrenal medulla, and the CNS, where they play a role in modulating autonomic and central nervous transmission. So clinically, drugs that target nicotinic receptors like succinylcholine primarily affect, okay, your NM, okay, your neuromuscular receptors, inducing paralysis by prolonged depolarization at the neuromuscular junction. You have to understand that distinction between our neuromuscular junction, NM, and NN, our autonomic ganglia, our CNS, our adrenal medulla. Okay? Understand that distinction, because agents that interact with these receptors must be selected in order to avoid unwanted side effects. For example, succinylcholine acts specifically on those neuromuscular junction receptors, ensuring muscle relaxation during surgical procedures without directly affecting the autonomic ganglia. here's another close-up picture from Apex. So that neuromuscular junction receptor located here, 5 subunits, 2 alpha, 1 beta, 1 delta, 1 epsilon in the adult and fetal. It's going to be a gamma. Acetylcholine binds to the alpha subunits. This leads to opening of that ion channel, leading to depolarization and muscle contraction. These receptors are the primary targets for depolarizing muscle neuromuscular blockers like succinylcholine and are non-depolarizers. Contrast that with your NN receptors. These are found in the autonomic ganglia and the central nervous system. Their subunit composition can vary, but they primarily consist of alpha and beta subunits. These are critical for the transmission of signals between neurons in the autonomic nervous system, and they can influence heart rate, blood pressure, and digestion. The distinction here between these receptors is important for drug selectivity, as our neuromuscular blockers target those NM receptors, while certain ganglionic blockers target those NN receptors to modulate autonomic activity. Okay, so NM receptor composition, 2 alpha, 1 beta, 1 delta, 1 epsilon, except in fetal, and then that epsilon is a gamma. Okay, NN receptors, autonomic ganglia, CNS, part of neuronal communication. Neuromuscular blockers target the NM receptors, okay, while ganglionic blockers target the NN receptors. So, plasma cholinesterases, they're made in the liver, found in the plasma. They're responsible for the rapid hydrolysis of succinylcholine. It's a very efficient process, so efficient that only about 10% actually makes it to the neuromuscular junction. And this is where it's going to exert its effect by causing muscle paralysis. The majority of the drug is broken down before it even gets there. What's it broken down to? Succinylmonocholine and choline. Succinylmonocholine has a much weaker neuromuscular blocking effect, and this is why it has such a short duration of action. And this process continues with succinylmonocholine broken down even further into succinic acid and choline, and these are actually inactive compounds. The rapid metabolism is a key feature that makes it very useful in situations requiring short-term muscle relaxation, like intubation. However, those with acetylcholinesterase deficiency, this breakdown is delayed. This can lead to prolonged paralysis, and you have to be very careful with these patients. So, hydrolysis of succinylcholine by acetylcholinesterase or butyrylcholinesterase or plasma cholinesterase occurs in the plasma, where almost 90% of the IV dose is hydrolyzed before it reaches the neuromuscular junction. It has a limited time of action at the receptor, okay, and it must redistribute back into the plasma via a concentration gradient that will facilitate redistribution and hydrolysis of succinylcholine. Now, cholinesterases, these are enzymes that play a crucial role in the breakdown of acetylcholine and other ester-bound drugs. Acetylcholinesterase, okay, is going to be at the neuromuscular junction and is responsible for the rapid hydrolysis of acetylcholine into choline and acetate, terminating the neurotransmission and allowing relaxation. This is a very highly specific enzyme to acetylcholine, and it's sometimes referred to as true cholinesterase. And its primary function is to maintain proper synaptic transmission by clearing acetylcholine from the neuromuscular junction, and it has after it has stimulated the muscle. So acetylcholinesterase, true cholinesterase, specific cholinesterase, genuine cholinesterase, type 1 cholinesterase. Okay? High quantities at the neuromuscular junction. Now, pseudocholinesterase, also known as butyrylcholinesterase or plasma cholinesterase, false cholinesterase, type 2 cholinesterase, serum cholinesterase, non-specific cholinesterase, benzylcholinesterase. It's found in the plasma, and it's less specific than acetylcholinesterase. It breaks down several drugs, including succinylcholine and some ester-based local anesthetics, which we'll learn about here in a little bit, like dibucaine. So this enzyme, pseudocholinesterase, is part of that rapid breakdown, which is why it has such a short duration of action. But you can have a deficiency. This can be genetic or acquired, and this can lead to a prolonged effect. Now, reversal of neuromuscular blockade is essential to gain back and restore your normal muscle functioning after surgery, especially if you're using a non-depolarizer. We have 2 primary types of reversal agents, okay? Neostigmine, which is acetylcholinesterase inhibitor, and then we have this selective relaxant binding agent like sugammadex. Acetylcholinesterase inhibitors work by inhibiting the breakdown of acetylcholine, increasing its concentration at the neuromuscular junction. This is competitive antagonism. Okay, this allows acetylcholine to outperform the non-depolarizing blocker at the nicotinic acetylcholine receptors, and this reverses muscle paralysis. Sugammadex, on the other hand, it's relatively new. All right, even I feel like I'm old saying this, but whenever I trained, I used Sugammadex literally like 3 times in training. All right, everything else I use neostigmine. So Sugammadex, it works specifically on our steroidal. We'll talk about that in a little bit, neuromuscular blockers. But a way to remember that is notice vecuronium, rocuronium, pancuronium, RON. Okay, just remember RON's on roids. Okay, those are your steroidal neuromuscular blockers. So Sugammadex encapsulates that drug. It renders it inactive and allows rapid reversal without those side effects of our acetylcholinesterase inhibitors. Sugammadex is very useful where you want a rapid and complete reversal. Think about an emergency or a deep blockade. Okay, the type of reversal you use, it depends on the neuromuscular blocker you've used. Sugammadex does not work on cisatracurium or atracurium or mivacurium. Okay, 99% of the time we're probably going to be giving Sugammadex because that's actually a recommendation. It's a recommendation because it is a better reversal. It's a true reversal. It's not competitive antagonism. Acetylcholinesterase inhibitors like neostigmine, they block the action of acetylcholinesterase, and this leads to a buildup of acetylcholine. Now, when you get this increased concentration, this will enhance neuromuscular transmission by prolonging its action on our nicotinic receptors at the neuromuscular junction. Okay, these are commonly used to reverse our non-depolarizers like ROC and VEC, cisatracurium, atracurium, to restore normal muscle functioning. By increasing acetylcholine levels, acetylcholinesterase inhibitors compete with the effect of our non-depolarizing agents, and this promotes the reactivation at the neuromuscular junctioning. But you have some clinical conditions, take myasthenia gravis for example, where there's a reduction in functional acetylcholine receptors. So those inhibitors improve muscle function by increasing the amount of acetylcholine available to activate the remaining receptors. However, careful dosing is critical. Excessive acetylcholine can lead to cholinergic crisis, cholinergic side effects, bradycardia, increased secretions, muscle weakness. That's why we give an anticholinergic along with it. Okay, acetylcholinesterase inhibitors, like neostigmine, it's truthfully a poison. Okay? Very similar drugs in that same family are literally nerve agents, are toxic. So when we talked about the old way to reverse paralytics with neostigmine and Robenal or glycopyrrolate, we would often say we give the antidote along with the poison, our anticholinergic, to help prevent those side effects. Succinylcholine-induced neuromuscular blockade can be significantly prolonged if someone has an abnormal genetic variant of pseudocholinesterase. So when this variant was discovered, it was noted that it responded to dibucaine, okay, and that's an ester-based local anesthetic, differently than it did to normal pseudocholinesterase. Dibucaine inhibits normal pseudocholinesterase to a far greater extent than the abnormal enzyme. So this led to an establishment of a dibucaine number. So dibucaine inhibits the normal enzyme by up to 80%, but the abnormal enzyme by approximately 20%. And there are a lot of other genetic variants. But oftentimes in patients with pseudocholinesterase deficiency and true deficiency, they'll have a dibucaine number. Okay? And that's what we will focus on. So this can be, pseudocholinesterase deficiency can be genetic, it can be acquired, and it really just affects the body's metabolism of succinylcholine and other ester-based local anesthetics, think dibucaine. This can lead to a significant delay in their breakdown. Okay? With this in mind, you have to be very careful. If someone says they're pseudocholinesterase deficient and you give them sucks, doesn't mean you're going to kill them, but it might mean you have to keep them intubated and sedated for a very long time. In the clinical setting, pseudocholinesterase deficiency often presents when a patient fails to recover from blockade, and the diagnosis is actually going to be confirmed by a blood test measuring pseudocholinesterase activity and that dibucaine number, that inhibition test. But if you have prolonged paralysis, you know it, and you use sucks, you know what happened. They might need to be mechanically ventilated and sedated until it wears off. because there's not a specific reversal agent for succinylcholine. Now, you'll see this, okay? If you do anesthesia long enough and use a lot of succinylcholine, you're going to see this at some point, okay? This happened to a colleague of mine who was a CRNA. When she was younger, she got put to sleep. She woke up hours later after her surgery and had been intubated and sedated because she was pseudocholinesterase deficient. So that can happen. That's a real thing. You're going to see the stats here in just a minute. There are some neuromuscular disorders you need to be aware of. Disorders like myasthenia gravis or maybe Lambert-Eaton syndrome, these actually impact the neuromuscular junction, and this can be by a reduction in the function of our acetylcholine receptors or an actual impairment of the release of acetylcholine. These conditions result in muscle weakness and fatigue, as acetylcholine is essential for muscle contraction. It cannot effectively stimulate the receptors. Myasthenia gravis reduces the number of receptors, while Lambert-Eaton syndrome reduces acetylcholine release. So these disorders are actually often treated with acetylcholinesterase inhibitors, which either will prevent the breakdown of acetylcholine or with immunosuppressants to reduce the autoimmune response in these conditions. So pseudocholinesterase deficiency is that genetic disorder characterized by prolonged neuromuscular blockade following the administration of succinylcholine. There's over 60 mutations affecting the gene coding for pseudocholinesterase, and most are rare, very rare. In a clinical scenario, the deficiency will become apparent when a patient is unable to breathe after surgery. They have a prolonged paralysis post-succinylcholine. And one reason why I avoid sucks, there's not a true reversal for it. However, management of the patient is placing them on a ventilator, allowing the drug to wear off, sedating them, okay, because they can be awake and paralyzed. And administration of fresh frozen plasma or human cholinesterase has been considered, but rarely is that ever given. You normally just let sucks wear off. And normally you want to have a dibukane inhibition test performed afterwards, okay? So, neuromuscular disorders. Myasthenia gravis affects the neuromuscular junction, reducing acetylcholine receptor activity or release, leading to muscle weakness. This is treated with acetylcholinesterase inhibitors. Pseudocholinesterase deficiency will prolong the paralysis after succinylcholine administration, requiring ventilator support and sedation. And you want that dibukane inhibition test, which will assess that pseudocholinesterase functioning. Now, the dibukane number. This is the percentage of normal pseudocholinesterase that is inhibited by dibukane. In patients with normal, the homozygous typical pseudocholinesterase gene, dibukane will inhibit 70 to 80% of this enzyme, resulting in a normal dibukane number. So normal, 70 to 80%. These individuals have a normal response and no significant prolongation of the neuromuscular block. There are two other variants. You have heterozygous atypical, okay, and they have a dibukane number of 50 to 60, meaning only about half of their pseudocholinesterase is inhibited by dibukane. And these individuals get a 50 to 100% increase in the duration of succinylcholine's effects. To contrast that with patients with a homozygous atypical pseudocholinesterase deficiency have a dibukane number somewhere in the 20 to 30 range, indicating significant deficiency. They may have a prolonged blockade lasting 4 to 8, up to 24 hours after succinylcholine administration. So prolonged blockade, think that homozygous atypical. Heterozygous atypical, okay, it's going to be about double the time for metabolism, so 20 to 30 minutes for your succinylcholine to go away, and homozygous is that normal 5 to 10 minutes. Now, this is just a picture to recap some drugs, okay? Drugs that are broken down by pseudocholinesterase, okay? Succinylcholine, neostigmine, edrophonium. These drugs all undergo rapid hydrolysis in the plasma, which accounts for their relatively short duration of action. Now, our non-specific plasma esterases. We haven't talked about them in depth, but atracurium and cisatracurium, romifentanil, clevidipine, etomidate are all metabolized by non-specific plasma esterases. These are esterases that are floating around in our plasma, which do not require the specialized enzymatic process from the liver. This allows for metabolism of these drugs even in patients with hepatic dysfunction. Romifentanil is unique because it has that ultra-short half-life, and that's because of that rapid metabolism. And then you have, and I mentioned it earlier, those red blood cell esterases. Esmolol is broken down by non-specific red blood cell esterases. And that's why it has such a rapid metabolism. Goodness, I love Esmol. Okay, if I want to beta block somebody in the OR, and I don't want to marry it, I don't want to be stuck with it for hours and hours, Esmol's great for short-term management. I'll get on my soapbox about long-acting antihypertensives have no place in intraoperative anesthesia. So these are just some drugs and the enzymes that break them down. Now the dibukane inhibition test outcomes. You can have a low dibukane number and lower activity, and this is an atypical enzyme with some prolonged apnea. You can have a normal dibukane number, but low enzyme activity. And this will suggest that while the enzyme is typical in form, there's lower levels of it. And this can still result in prolonged apnea due to the insufficient enzyme quantity. Now, a low dibukane number with a very low activity suggests the presence of a rare variant type. Okay, significantly impaired functioning, very low levels, severe and prolonged apnea of succinyl choline. Now, normal dibukane number and normal activity, okay, normal enzyme and amount, there would be another reason for prolonged apnea, if that is the case. Although succinyl choline does not affect the CNS directly, it can cause a transient increase in ICP, usually in the range of 10 to 15 millimeters of mercury, lasting for about 5 to 8 minutes. And this is due to the increased cerebral blood flow, muscle activity, and fasciculations in the neck muscles, which can contribute to jugular vein stasis and increased pressure. The rise in ICP is of critical concern in someone with a head injury or an intracranial lesion, or maybe has cerebral compromise, where even small changes could exacerbate a clinical outcome. To mitigate this, we often use a defasciculating dose to stop those fasciculations, okay? And what that is that's a small amount of a non-depolarizer before the administration of succinyl choline. And this is normally with 10 to 30% of the ED95 of the non-depolarizer, okay? Normally 5 to 10 milligrams of rocuronium, and sometimes, a milligram or two of vecuronium. By using this approach, we can minimize those hemodynamic changes that succinyl choline causes. So this way they do not have those fasciculations, okay? Because the fasciculations are associated with the increase in ICP. So just a couple things to remember. General capillaries are fairly permeable, okay? They are going to allow substances to pass through. Lipid-soluble substances also readily diffuse across capillary walls, making it easier for most drugs and molecules to enter tissues throughout the brain. Our brain capillaries and our blood-brain barrier, these are capillaries within our brain that are protected by that barrier, okay? That highly selective barrier is formed by tight junctions between endothelial cells. These prevent large, charged, or non-lipid-soluble substances from passing into the brain. Instead, only lipid-soluble substances and small molecules can diffuse through the blood-brain barrier, okay? That's just something to keep in mind as we're talking about this. Drugs like succinyl choline cannot cross the blood-brain barrier because of its charge, its poor lipid solubility, and the size of the molecule, okay? That's why the effect is limited to the peripheral tissue. Postoperative myalgias are a very well-known side effect of succinyl choline, especially in the neck, face, abdomen, shoulders, and trunk. And these are the result of fasciculations, which are caused by that uncoordinated repetitive firing of our motor nerve terminals at our neuromuscular junction. This can lead to short, uncoordinated contractions, and these produce soreness and discomfort after the paralytic wears off. These are more common in women and individuals who are less physically active, sedentary, okay? Certain populations, including the elderly and pregnant people, along with very young patients, tend to be more resistant to myalgias. To mitigate myalgias, we sometimes use a defasciculating dose of a non-depolarizer, okay? And this is a small dose of a non-depolarizer before succinyl choline, okay? This reduces the intensity and frequency of a fasciculation and decreases the likelihood of postoperative myalgias. Although it cannot guarantee for 100% that you won't have myalgias. Succinyl choline has some cardiovascular effects, okay? It can cause a little slight tachycardia, and this can be from a little sympathetic stimulation, but there can be incidences of sudden and abrupt bradycardia, especially in adults with repeated doses or any dose given to a pediatric patient. This bradycardia is primarily attributed to the stimulation of muscarinic receptors in the parasympathetic nervous system or through succinylcholine, which can activate these receptors at the SA node. You have to keep this in mind because this is crucial for managing anesthesia in the pediatric population. There are also some renal considerations. Okay, patients with renal disease may exhibit lower levels of cholinesterase. Okay, lower levels of pseudocholinesterase. This can lead to prolonged neuromuscular blockade. Succinylcholine can also increase serum potassium levels. This can pose a risk for hyperkalemia. Therefore, it's essential to assess preoperative potassium levels in those with renal issues to prevent potential complications from this. You have to be aware of it. Can you use succinylcholine in someone on dialysis? Yes. Okay. As long as they don't have something else that would preclude it and their potassium level is within a somewhat normal range. So, no contraindication of the use of succinylcholine exists in obese patients. Okay, the recommended dose of succinylcholine is actually 1 milligram per kilo. Okay, I'm not talking about an RSI dose, but this is based on total body weight. Okay, and that will give you excellent intubating conditions. Because succinylcholine is water soluble, it would seem that doses based on lean body weight would suffice. However, morbidly obese patients have increased fluid compartments and pseudocholinesterase levels, and therefore require a higher dose to get paralysis. Now, succinylcholine has some influence on the gastric physiology, okay, because it can increase both intragastric and lower esophageal sphincter pressure. This elevation can lead to complications during anesthesia, such as an increased risk of aspiration. The use of a defasciculating dose may help mitigate this issue. In our elderly patients, the onset of succinylcholine may be slightly prolonged due to those age-related physiologic changes and the impact of medications that could directly inhibit cholinesterase enzymes. Okay, diazepam, okay, rivastigmine, galantamine, those are all medications that a patient might be on. All right, the dosage, once again, should be on total body weight to ensure that there is an adequate effect, okay, even in morbidly obese patients. All right, this is essential, okay, for adequately dosing succinylcholine. I talked a little bit about the intracranial pressure, but succinylcholine can also increase intraocular pressure. This is somewhere in the range of 5 to 15 millimeters of mercury by up to about 10 to 15 minutes. This is related to the vascular dilation or potentially decreased drainage secondary to elevated CVP. And this can inhibit the flow of aqueous humor, and this is certainly good to note for someone who has elevated intraocular pressure. Okay? You have to remember, though, it cannot be blunted by using a defasciculating dose. Okay? This is very concerning with someone with a pre-existing eye condition. While there have been no reports of intraocular globe contents extruding, okay, the risk remains a potential concern, okay, particularly in cases where securing the airway is vital. Therefore, carefully assess that patient, okay? On the off chance, just avoid sucks, okay? If it's warranted, maybe do an RSI with rock. I mean, coughing and bucking can also increase intraocular pressure, okay? Especially if they're bucking on the vent, this can actually increase it probably more than sucks, but you have to keep this in mind. That is a potential theoretical risk. Okay. Additionally, patients with conditions that result in a proliferation of extrajunctional acetylcholine receptors, think upper and lower motor denervation, immobilization, someone who is immobile, burn injuries, neuromuscular disease, they're likely to have an exaggerated hyperkalemic response following administration of succinylcholine. So, when the frequency of stimulation at the neuromuscular junction decreases over days or longer, think about severe burns, immobilization, infection, sepsis, prolonged use of neuromuscular blocking agents in the ICU, CVA, stroke. The number of immature nicotinic acetylcholine receptors increases. You have receptor upregulation, and they extend beyond the neuromuscular junction into the adjacent muscle membrane. These immature receptors have increased sensitivity to agonist, acetylcholine and succinylcholine, and decreased sensitivity to non-depolarizers. The channel opening time of the immature receptors is up to 10 times longer than that of the mature receptors and can allow for systemic release of lethal doses of intracellular potassium. Downregulation of mature receptors occurs during periods of sustained agonist stimulation. So chronic neostigmine use in someone with myasthenia gravis or organophosphate poisoning can lead to resistance to succinylcholine, but extreme sensitivity to non-depolarizers. Okay. The use of SUX is contraindicated in several clinical scenarios due to the heightened risk of many adverse effects. Hyperkalemia. Succinylcholine can elevate potassium levels. It's contraindicated in patients with extensive burns, think third degree or greater than 35% body surface area, severe muscle trauma, neurologic injuries like paralysis or quadriplegia, okay? Muscle wasting, malignant hyperthermia, Duchenne's muscular dystrophy, any muscle disorder, okay? Probably avoid succinylcholine. In pediatrics, we normally use SUX as an emergency drug, okay? It's part of airway management due to the risk of severe complications, especially in undiagnosed cases of muscular dystrophy, which can lead to hyperkalemia, which can lead to a fatal outcome. It's crucial to carefully assess the patient's history and current conditions to avoid administering succinylcholine. Keep these in mind. If they're immobile, don't use SUX, okay? If you think, well, maybe I shouldn't, don't use SUX. Be safe. In children, succinylcholine is only used in emergency situations to secure an airway. Routine use in elective procedures was abandoned in the early 90s. This is because there were some widely reported cases of severe hyperkalemia and rhabdomyolysis in what appeared to be healthy children. These cases involved routine procedures in children with actually undiagnosed Duchenne's muscular dystrophy. So this is an X chromosome-linked disorder with an onset of symptoms usually around 5 years of age. And patients will typically get a progression of weakness and atrophy that starts in the legs and pelvis and can spread to the shoulders and neck. And this can involve the upper extremities and respiratory muscles. Life expectancy is rarely more than 30 years of age, and death is a consequence of that cardiac and respiratory disease. So children with Duchenne's muscular dystrophy frequently require orthopedic surgery for repair of scoliosis or contractures. So there's a black box warning, okay, for the risk of cardiac arrest from hyperkalemia, rhabdomyolysis, okay? You're normally going to see this, peaked T waves and sudden cardiac arrest within minutes after the administration of SUX. This is why it's so important to only use succinylcholine in an emergency in kids. Therefore, when you have a healthy appearing child or infant and they develop cardiac arrest, and it's not due to inadequate ventilation, oxygenation, or anesthetic overdose, immediately treat them for hyperkalemia. IV calcium, bicarb, glucose with insulin, hyperventilation. Routine resuscitative measures are likely to be unsuccessful if you do not understand this, okay? It is important to note that extraordinary and prolonged efforts have resulted in successful resuscitation, okay? And in addition, you may have signs of MH. It's just best to avoid it, okay? Emergency situations only, okay? In pediatric patients, it's great for, a laryngospasm, but you have to be very, very cautious because of these risks. So the increase in potassium from succinylcholine, so with the activation of acetylcholine channels, movement of sodium into the cells is accompanied by movement of potassium out of the cells. Okay, patients with renal failure are actually no more susceptible than those with normal renal function. So you can use SUX in renal disease as long as nothing else precludes you from using it. The response of patients with neuromuscular diseases, like CVAs, hemiplegia, paraplegia, muscular dystrophies, Guillain-Barré, be very, very cautious. It could be severe, it can lead to cardiac arrest. Okay? So, succinylcholine can cause a transient increase in serum potassium shortly after administration. Typically, the levels increase by 0.5 to 1 milliequivalent within 3 minutes, lasting approximately 10 to 15 minutes. Prolonged depolarization associated with succinylcholine allows potassium to leak from muscle cells into the bloodstream. This can pose a risk for hyperkalemia, especially in those with a pre-existing condition. In healthy patients, acetylcholine receptors are localized in our junctional areas, okay? And you normally are not experiencing that. However, Conditions like denervation, infection, trauma, immobilization, there's an upregulation of these receptors throughout the muscle membrane. This can lead to that exaggerated potassium release. In burn patients, this upregulation occurs within 24 to 48 hours post-injury. Okay? So if it's one day after the burn, just don't use it. Don't use sucks. Malignant hyperthermia is also triggered by succinylcholine. This is that rare but life-threatening condition that can be triggered by our volatile anesthetics and sucks. Okay? This is a hypermetabolic state due to uncontrolled calcium release from our sarcoplasmic reticulum. This leads to sustained muscle contractions, elevated heat, and this is all from a genetic predisposition, and it's normally a mutation in the ryanodine, the RYR1 receptor. Okay? Those individuals are at high risk for developing this syndrome. Anybody who has a family history of MH or MH, avoid sucks. The presence of malignant hyperthermia is an absolute contraindication for the use of succinylcholine or volatiles. You have to be vigilant for signs of MH. Okay? Tachycardia. Elevated end-tidal CO2 that's resistant to changes in minute ventilation. Muscle rigidity. Metabolic acidosis. Temperature elevation is a late sign. You have to get early detection. Okay? Proper patient history is essential. If they tell you, oh, I think I had a family member who had problems with anesthesia, do a TIVA. Do a TIVA, do a TIVA. Be safe. All right? Avoid sucks. One other reason why I avoid it the majority of the time, it can unknowingly trigger MH. MH is a pharmacogenetic skeletal muscle disorder, and it's triggered by your volatiles and succinylcholine. Succinylcholine is absolutely contraindicated along with volatiles in someone with a known or suspected MH history or has MH in their family. Okay? Mutations in that RYR1 receptor can result in altered calcium release from the sarcoplasmic reticulum, causing cells to lyse, potassium to break out, increased CO2 production. This can lead to convulsions, acidosis, tachycardia, and then the hyperthermia is a late sign. Okay? Avoid it, avoid it, avoid it. Okay? Signs of malignant hyperthermia. Okay? Elevated temperature, muscle rigidity, tachycardia, metabolic acidosis, elevated end-tidal CO2, myoglobinuria. All those things, okay, are signs and symptoms. If you are wondering, should I treat for MH? Is this MH? They have to have at least 2 and have had a triggering agent. And if you decide to go that route, you have to go all the way. You have to give your dantrolene, and if you haven't had, some resolution of symptoms, it's probably not MH. Okay, so now let's talk a little bit about a depolarizing and a non-depolarizing block. Okay? Neuromuscular blockade is categorized into depolarizing and non-depolarizing, and they each have some distinct characteristics. In a depolarizing phase 1 block, succinylcholine is commonly used. This is characterized by initial fasciculations, which precede the blockade. Sustained muscle contractions result in a lack of fade during tetanic stimulation. Okay, so our phase 1 block. So if this is, we're going to draw this. Oh, let me get this here. We're going to draw this, okay. We're going to train of 4, TOF. Okay, so we're going to have 4, okay, 4 bursts. Okay, normal train of 4 and mirror you right now. This is separated by a 0.5 second apart. These lines should all be about the same. So this is the response right here. Okay, so how high it is that response. Okay, sustained muscle contraction results in a lack of fade during tetanic stimulation. Okay, and there's an absence of post-tetanic potentiation. A notable feature of a depolarizing block is that it can be potentiated by anticholinesterase drugs, and its effects can be antagonized by the prior administration of non-depolarizing agents, requiring approximately 20% more succinylcholine for effective blockade. Okay, so if this is normal train of 4, okay, when you give succinylcholine, this is what will happen. Okay, this is your initial twitches. Okay, you're stimulating, boom, 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 boom. They're all the same. Okay, then they're going to be less. okay, you're going to have less of a response if you do the train of 4. And finally, it's going to be minimal to none, okay? And I said that word fade, okay? Contrast that. A non-depolarizing, a phase 2 block, you don't have fasciculations, okay? It presents with a typical tetanic fade and post-tetanic potentiation, with the train of 4 response demonstrating a decline, okay, in muscle response. This type of block can also be reversed using anticholinesterase drugs, and high doses of succinylcholine can produce a phase 2 block. Keep that in mind, high doses of sucks, greater than 6 milligram per kilo. They used to do suck strips, no one does it anymore. Okay, that can give you a phase 2 block. Understanding this is critical for effective management of neuromuscular blockade and for anticipating potential complications. So, phase 2 block. This is normal. So this is our normal 1, 2, 3, and 4. Okay, that's normal. Okay, as it sets up, your first twitch, your second twitch is a little weaker, your third twitch, and then your fourth twitch is very, very weak. Okay, and then you may have 1, 2. Okay, so the first twitch, second twitch, and you have no response there, and then eventually you'll have no response. Okay, that's fade. It's fading. Okay, fading. Okay, there's no fade here. Okay, that should be at the top. No fade. All right, this is fade. And we're going to get into this more later, but just to talk about it before we get into reversal agents, just so you can remember that sucks can cause, and normally causes, a phase 1 block, okay? Fasciculations, sustained tetanic stimulation, no post-tetanic potentiation, no fade. It can cause a phase 2 block, but normally you get a phase 2 block from your non-depolarizers, and no, hopefully you're not giving greater than 6 milligram per kilo of sucks. Don't do that. All right, I mentioned this earlier, but our non-depolarizers, they're broken down into 2 categories. Our steroidal, remember, let me mark it up for you now that I'm in the marking phase of the presentation. Ron is on roids. Okay, steroidals. Vecuronium, rocuronium, pancuronium. These are competitive antagonists. They inhibit the action of acetylcholine, resulting in muscle relaxation. Okay, you only need them down to 1 alpha subunit. Now your benzyl isoquinolines, okay, atracurium, cisatracurium. These are another class, okay? Sugamidex does not work on these, okay? Let's just do Sugamidex. Sugamidex here. Nope, doesn't work. What do you use? Neosigmine. Ugh. Okay, they're truly falling out of favor. Now, cisatracurium or Nimbex, you're probably familiar with it from the ICU days. You use it as an infusion. But so they're non-depolarizers. They undergo spontaneous degradation and are metabolized through plasma and tissue esterases, and they can be suitable for renal or hepatic impairment. And there's utility behind them, but the majority of the time we're going to be going with our steroidals. You're probably going to see Rockenbeck. Like I said, I've never even given pancuronium. Certainly teach about it, talk about it, but it's not one you see very often. So now let's jump into pancuronium. This is a very potent, long-acting neuromuscular blocker. It's steroidal, and pancuronium is known for a couple very specific effects. It has a vagolytic and a sympathiomimetic effect. It will block the reuptake of norepinephrine and inhibit butyrylcholinesterase, and this gives you a prolonged neuromuscular blockade. It's very useful. Think heart surgery. It's something that's going to be very, very long. As far as metabolism go, 40 to 60% is cleared by the kidneys, 11% in the bile, a small percent, 20% undergoes metabolism by the liver, and its metabolites are less potent, all right, and are eliminated in the urine, okay? Rarely used, okay, but if you're going to use it, big cases. Okay, one key concern for pancuronium is that prolongation of neuromuscular blockade, especially the accumulation of that 3-OH metabolite. This can lead to extended muscle paralysis and can pose a risk of patients after surgical procedures. you have to be vigilant about it if you're going to be using pancuronium. Additionally, okay, pancuronium, it is not well reversed by Sugammadex comparatively. Sugammadex is designed for rock uronium, and it will reverse all your steroidals, but it works for rock the best, VEC the second best, pancuronium the third best. Okay, so it's not as strong. Okay, so oftentimes we're using a shorter acting agent, and also you give pancuronium, you're kind of marrying it. All right, that's another thing. You're getting a very long muscle relaxation. Okay, so pancuronium has those vagolytic effects, okay, as well as that direct sympathomimetic effect. It blocks norepinephrine presynaptic reuptake. Okay, it's highly potent, so that means it has a slow onset. All right, so you give it, you're gonna have to ventilate for a little bit. Okay, it's slow onset, so doses greater than 2 times our ED95 are usually needed for intubation in less than 5 minutes. So you're gonna have to give quite a bit. Okay, pancuronium historically, we love it in cardiac surgery because that vagolytic effect counteracts the bradycardia with a high dose narcotic technique. You give a bunch of narcotic, that's the old school way of doing hearts. 20, 30 cc's of fentanyl, pancuronium, you don't get that severe bradycardia from those opioids. All right, but there's that downside. Okay, you can use Sugammadex, but it's not as effective. This is, if something happens where you have to wake a patient up afterwards, pancuronium, you're marrying it. You give it for intubation, gosh, it's going to take forever. And if you give one neuromuscular blocker, if you give rock, you don't switch to pancuronium. You can go rock to VEC, VEC to rock, but no one's really using pancuronium. But you need to know about it, okay? Elimination, renal pathways, a little broken down into the liver. Have to be very careful on those with kidney disease as far as like that prolonged paralysis. Okay, vecuronium. Okay, this is actually derived from pancuronium. Okay, it has some distinct properties that make it very, very nice. Okay, it has a molecular structure differing from pancuronium in the absence of a cortinizing methyl group, and this results in a couple changes in our pharmacological effects. Vecuronium has a decrease in potency compared to pancuronium, and this can affect our dosing strategies. We also have no more vagolytic properties. Okay, we have increased lipid solubility. Now we get greater biliary elimination. It has molecular instability in solution. Therefore, vecuronium comes to you in a powder, and you reconstitute it, normally with sterile water. Okay, so normally you're going to get a 10 or 20 milligram vial of powder, and then you're going to reconstitute it. Okay, normally vecuronium is reconstituted as 1 milligram per ml. Okay, so you put 10 cc's of sterile water, you mix it up, you pull it back. Now, can you do it concentrated? Can you do 2 milligrams per ml? Sure, you can. I do that normally. I'll take a 5 cc thing of sterile water, squirt it in my VEC. It works great. Okay, so differences: decrease in potency, no vagolytic properties. It has molecular instability, so it's going to be in that powder, and you get increased lipid solubility. As far as elimination pathway goes, primarily eliminated by the liver, 30% cleared through the kidneys, and you do have 30 to 40% excreted in the bile. Now, as far as metabolism go, okay, it undergoes metabolism in the liver through a process of deacetylation, and you have 3 different metabolites. That 3-OH metabolite still has about 80% of VEX potency, and this can complicate the use of VEC, okay, because this metabolite is crucial for you to keep in mind. The duration of action of VEC is influenced by our hepatic function, and clearance is dependent on liver metabolism. Renal functioning can also play a role, but to a lesser extent. Once again, it's in a powder, and you have to reconstitute it. Okay, so longer duration of action than rock uronium. Okay, if you give it up front, for intubating, once again, it's fairly potent, so it has a shorter onset. All right, but you can still intubate on VEC, but you're gonna have to ventilate 3 or 4 minutes. And normally an induction dose of VEC, all right, 0.1 milligram per kilo, 0.1 to 0.15, and if it's gonna be a long case, 10 of VEC up front works great. For heart, sometimes you'll give 20 of VEC. It's a long surgery. If you're wanting quicker extubation times are probably going to give rock, but vecuronium is, a lot of times used in cardiac anesthesia or very, very long cases. Okay, it's a potent agent that has slight lipophilicity. Okay, once again, it's hepatically eliminated. So once again, broken down to those compounds, it's fairly potent, okay, compared to rocuronium. 10 or 20 milligram powder reconstituted to 1 to 2 milligrams per ml. Maximal twitch depression is normally in about 3 minutes, okay, with effects lasting 30 to 60 minutes, maybe on that 90 side if you're on that higher dose, or if you have any clearance issues. All right, 30% of the drug is unchanged in the bile, and we do have that active metabolite, okay? So with that in mind, okay, vecuronium is ideal in cases where it's going to be 2 hours plus, and it can be titrated. The historical teaching is vecuronium was more predictable. Rocuronium it gets a bad rep for being unpredictable. All right, how long is it going to last? Who knows? All right, Vec for the longest time was more predictable, and they would say it was quote unquote easier to reverse with neostigmine, even though that metabolite hangs around. So you're still going to see it in your trays. Some people are still going to use it. You can reverse with Scamodex, but a lot of times if you're using a non-depolarizer, it's going to be rock. All right, this is our intermediate acting drug. Okay, faster onset compared to vecuronium and pancuronium. This is very valuable when you want to RSI. You can RSI with rock. Perfect. It's just a little slower than succinylcholine. Okay, rocuronium is about 1/6 as potent as vecuronium. Okay, that's why you have to give a larger dose of it. So in terms of elimination, okay, processed through the liver, excreted in the bile. Rocuronium is stable. Okay, it's stable at room temperature for up to 60 days. All right. Faster onset. 6 times less potent. All right, so 1/6 the potency. All right, stable at room temperature. Now, rocuronium is metabolized in the liver. Its metabolic pathway results in a small amount of a metabolite, which has really not been detected in significant amounts. So it's fairly good. Okay, it's clinical use, fast onset, intermediate duration. Okay, you want to remain cognizant of that hepatic functioning. That's why we're not really using rocuronium drips much in the ICU. We're not. Same thing with VEC drips. That's why I like Nimbex so much, or cisatricurium. But rock, we love it in the OR. We love it because Scamodex works so well to reverse it. Okay, so one method used to accelerate the onset of rocuronium is a priming dose. Okay, I don't do this personally. I've seen some people do it, but priming involves giving about 10% of your intubating dose prior to inducing. Okay, after a period of 1 to 3 minutes, the patient will still be anesthetized, and the rest of the rock is given. So giving a small amount speeds the onset pretty quickly. All right. The downside is this can produce stress, respiratory distress, and anxiety. Not a big fan of it. So what you do is while you're pre-oxygenating, a minute or two before you induce, you give that rock. Okay. Rocuronium actually burns when it goes in. Okay. If you ever, next time you're in the OR, drop a half cc of rock, squirt it on a cotton swab, smell it. It stinks. Okay. Rocuronium burns. Okay. The stabilizer within it. If I remember correctly, rocuronium has, not only is it the stabilizer that it's mixed in, which smells really bad, it also has a fairly acidic pH. So normally, if your patient's complaining of burning, you're probably giving propofol, but if you give that priming dose, they're probably going to complain of pain. Okay, so keep in mind how it is eliminated. So the elimination half-life, 60 to 120 minutes, depending on the dosage. So slightly longer onset time compared to SUX, but very useful in an RSI. 0.6 to 1.2 milligram per kilo can give you good intubating conditions. RSI dose is probably that 1.2. Rock is a derivative of VEC. Okay, 1/6 the potency. And once again, biliary excretion. Atracurium. Okay, atracurium and cisatricurium are not steroidal. They're benzoyl isoquinolines. So the metabolism is unique. It undergoes spontaneous degradation through Harmon elimination, and there's a temperature and pH dependent process that produces londanicine, okay? And this is important. We're going to talk about it here in another slide or two, but atracurium is suitable for patients with compromised hepatic or renal functioning, as its breakdown is not on the traditional metabolic pathway, okay? But atracurium causes some not so great side effects. One effect is histamine release, okay? So a little bit more about this londanicine and Harmon elimination. Okay, Harmon elimination is a metabolic process for breaking down atracurium, okay? It involves the breakdown of the drug at a physiological pH and temperature. That's why you have to keep them warm, have to keep them, keep their pH normal, okay? This reaction yields londanicine and a monoquaternary acrylate. And these are all part of atracurium's pharmacokinetics. And about 70% of that londanicine is excreted via our bile, while the remainder is cleared through the urine. Clearance of this can be impaired if you have a biliary obstruction or is unaffected. Londanicine can cross the blood-brain barrier and it can cause some CNS side effects. Okay, current evidence suggests that there is really no significant risk of seizure associated with atracurium use in patients with normal or impaired renal functioning. But the plasma elimination half-life of londanicine is 4 to 5 hours. Okay, and while it's a metabolite, you should monitor the CNS effects. No one's really using atracurium anymore. No one is. That histamine release, ugh, it's terrible. Okay, so aside from the londanicine, okay, which has the CNS stimulating properties, and londanicine crosses the blood-brain barrier. All right, atracurium loses potency over time. All right, that's why it's refrigerated. The duration of action is between 30 and 60 minutes. So quick on, quick off, causes histamine release. Causes histamine release. Atracurium causes histamine release. Remember that. Okay, this can lead to adverse cardiovascular effects like hypotension, tachycardia, flushing, changes in cardiac output, bronchoconstriction. Okay, the metabolism of it is through Harmon elimination. That's temperature and pH dependent, and then some non-specific ester hydrolysis, and then it has that metabolite londanicine, which can cause CNS activity. Okay, so, and londanicine is highly lipid soluble. No one's really using it. Cis-atracurium, what is this? It's an isomer of atracurium. Okay, it's a little bit more potent, 4 to 5 times as potent. Produces much less londanicine. Okay, and there's pretty much no histamine release. Okay, so its structure is an isomer of atracurium. It still undergoes Harmon elimination. Okay, but it does not undergo ester hydrolysis. And the clearance is majority Harmon elimination, so you just keep them warm, okay? Keep their pH normal, and it's fairly predictable. That's why you can run it as an infusion. So it has a unique property, okay? It does produce londanicine, but it's at a rate of about 4 to 5 times less than atracurium. Okay, so it's significantly reduced, okay? It decreases the risk of any accumulation issues or clinical consequences. That's why cis-atracurium is so nice in the unit. That's why you're using Nimbex in the unit. Histamine release is very common with atracurium. Histamine is pretty much none with cis-atracurium. Okay? With that being said, cis-atracurium has a very favorable pharmacological profile. Okay? Reduced londanicine production, absence of histamine release. It's preferred in patients with compromised organ functioning. Okay, although a lot of us are still using Roc and Sagamedex. Key point though, if you give someone Sagamedex and then immediately have to reintubate them, you can use Sux or cis-atracurium. I guess you could use atracurium, but if you've given Sagamedex, you're not going to use a steroidal. So that's another time that cis-atracurium might be useful. So 4 to 5 times as potent. All right, no histamine release. Still works through Harmon elimination. Can be given as an infusion. Much less londanicine production. Okay, this is why it is very good in those with compromised organ functioning. Now, mivacurium. Movicron. Ugh. Okay, I've never used this, but everyone, and this is coming back around. They're repurposing it. So mivacurium is another benzyl isoquinoline, and it's actually metabolized by acetylcholinesterase, okay? And rapid metabolism, short duration of action, okay? So this was great in patients who had, like a 20-minute ENT case. You needed quick relax and a quick cysto. It worked great. However, mivacurium, all right, wore off quickly. It causes histamine release. That can cause the hypotension and bronchospasm, okay? It's coming back around. Be very careful with it. If your facility gets it, I would be very, very cautious. That hypotension and bronchospasm, not a good thing. When we talk about the potency, this refers to the amount of drug required to achieve a specific effect, okay? And we normally measure this in the ED50 or the ED95. So potency is often expressed in micromoles per kilo. So for example, rocuronium has a low potency, therefore it has a rapid onset. And the reason why, okay, is since it has a low potency, you have to give a larger amount, okay? And what this does is this helps reach effective site concentrations quicker at the neuromuscular junction. Think about an RSI dose of rock, 1.2 milligram per kilo, 100 milligrams, okay? You may give 100 of rock. You may only give 10 milligrams of vecuronium, okay? So rocuronium can saturate, even though it's less potent, it has a quicker onset, can saturate those receptors. So the speed of onset is inversely related to potency. High potency, longer effect. Low potency, quicker effect. So rocuronium, low potency, quicker onset. That's why we like it in that RSI situation, okay? Vecuronium, cisatracurium, more potent than rock, slower onset. Low potency drugs have a greater number of molecules available, leading to a faster diffusion gradient, increasing the rate of drug transfer. Now, buffered diffusion is a phenomenon where highly potent neuromuscular blockers are impeded in their diffusion from the neuromuscular junction because they repeatedly bind to high-density receptors. And this can prolong the duration of neuromuscular blockade. And some drugs like curare exhibit this effect. Okay, so when you see that curare or D-tubocurarenine, it's a benzyl isoquinoline. This is relevant to that buffered diffusion, okay? Long-acting, slower recovery time. So the persistence of high-potency drugs results in a prolonged duration of effect. This is why you have to be very careful. So buffered diffusion occurs when you give a potent drug repeatedly and it impedes their diffusion. This can prolong and complicate that blockade and recovery. That's why oftentimes we really like rocuronium. We really, really like it. It's easier to reverse. You have a specific reversal, and it's less potent. Now, there are some factors that potentiate non-depolarizers. Let's start with our inhalational anesthetics. These have a significant effect. So this results in a reduction in the dose needed to achieve muscle relaxation, and they can prolong the drug's duration of action. And the degree of this potentiation depends on the specific inhalational agent used. Desflurane augments muscle relaxant the most, followed by SIBO, ISO, halothane. Nitrous, barbiturates, opioids, and propofol have a minimal effect as far as potentiation of these goes. Now, this mechanism includes both a central and peripheral effect. Our inhalational anesthetics act centrally by depressing our motor neurons and our interneural synapse, and thus reducing neural transmissions. They also have a little bit of an inhibition in our postsynaptic nicotinic acetylcholine receptors, and they increase the affinity of our non-depolarizers. So this leads to a stronger and longer lasting neuromuscular blockade. Okay, so with that in mind, all right, DES, SIBO, ISO, halothane. Okay, desflurane, sevoflurane, isoflurane, halothane. Nitrous, barbiturates, opioids, and propofol, minimal effect. There are some other drugs like antibiotics, are aminoglycosides, polymyxins, lincomycin, clindamycin. These can all potentiate the effects of our non-depolarizing neuromuscular blockers. And they do this by inhibiting acetylcholine release at the neuromuscular junction and reducing receptor sensitivity. They block calcium channels at the presynaptic nerve terminals, and this inhibits the release of acetylcholine, and that's essential for muscle contraction. Tetracyclines also enhance the effects of our neuromuscular blockers through post-junctional activity. Magnesium sulfate, hypothermia, these can also potentiate our non-depolarizers. Magnesium sulfate will inhibit calcium influx, reducing acetylcholine release, while hypothermia just slows that metabolism, prolonging the effects. You have to be aware whenever you're using all these things. So if you're doing a general anesthetic, you're using DES and you're using MAG and you're giving clindamycin, you're probably really potentiating your neuromuscular blocker. Now let's talk a little bit about hypothermia. So hypothermia significantly impacts the pharmacodynamics of our neuromuscular blocking drugs and increases their duration of action. This is very noticeable whenever you're using agents like vecuronium, where the recovery time to achieve a 10% twitch height can double when body temperature decreases from 36 to 34. And this involves slower metabolism and clearance at those lower temperatures. Keep your patients warm. Okay? Clinically, profound hypothermia can complicate the monitoring of our neuromuscular responses, especially when we're talking extreme levels like 27 degrees Celsius. Okay? At that point, standard neuromuscular monitoring may become unreliable. You have to be very vigilant in these cases. Okay? Don't let your patients get super cold. All right? Hypothermia can really increase your neuromuscular blockade. Okay, what about our local anesthetics? Some local anesthetics can significantly potentiate these as well, especially at high doses. Okay, this is by blockade of sodium channels, which are critical for our neuromuscular transmission. By inhibiting the propagation of action potentials along our motor nerves and muscle fibers, our local anesthetics enhance the effects of our non-depolarizers. Okay, this can lead to prolonged neuromuscular blockade, and this is very important when you consider surgeries where both local anesthetics and our non-depolarizers are used. Okay. Some anti-dysrhythmic drugs like quinidine can enhance the effect of non-depolarizers because quinidine inhibits sodium channels, similar to the local anesthetics, but also reduces the release of neurotransmitters at the neuromuscular junction, intensifying that blockade. Keep that in mind, okay? This all adds into careful monitoring. Okay? What are some factors that reduce the potency? Well, several factors can reduce the potency of our non-depolarizers. And this would mean we would need a higher dose to achieve adequate relaxation. Chronic anticonvulsant therapy, especially drugs like phenytonin and carbamazepine, they increase the clearance through enzymatic induction. Okay, those anticonvulsants increase the metabolism of our non-depolarizers and shorten their duration of action, and they need a larger, more frequent dose to maintain effective paralysis. So, I'll give you an example. You have a patient who's been on anticonvulsants because they have seizures and they're getting a craniotomy. I've given a large dose of rocuronium before, and within 30, 45 minutes, I see the patient moving. There's no way 4 twitches. And it took a lot of rocuronium to keep them relaxed. Hypercalcemia. Okay, this is also associated with hyperparathyroidism. This can decrease the sensitivity to our neuromuscular blockers. Elevated calcium levels enhance acetylcholine release. Okay, because acetylcholine is going to compete with that non-depolarizer for that receptor binding site, okay? And in these cases, you may have to adjust your doses to ensure adequate relaxation. Building on that, hypercalcemia can lead to a resistance in these drugs, okay? Calcium can have a huge effect on the anesthetic management, okay? This might mean that you need a higher dose. This might mean that you need to be checking your twitches much more frequently. And the reason behind this is calcium's role at the neuromuscular junction. Calcium will enhance the release of acetylcholine. This is going to stimulate muscle contraction. And by increasing those acetylcholine levels, okay, by increasing the acetylcholine release, calcium raises that threshold for neuromuscular blockade, shortening the duration, okay, decreasing the potency, okay. Clinically, hypercalcemia is known to reduce sensitivity, okay, to atracurium, curare, pancuronium, and this can all result in insufficient blockade. You have to plan for this. Now let's talk about some of our special populations. Now, the onset times of our neuromuscular blockers are generally delayed in the elderly due to slower circulation times and other kinetic changes associated with aging. Okay, this is typically true for all relaxants. The dosing interval and the duration of action for steroidal relaxants like Rockvec and pancuronium are prolonged in the elderly due to decreased hepatic and renal clearance. Atracurium and cisatracurium, the duration is not really affected as much by aging. They're a little bit more predictable. But we're normally going to use rocuronium or vecuronium to use Sagamedex. Now, contrast that with the neuromuscular junction is incomplete at delivery, so in infants, and it matures throughout infancy. The sensitivity to any relaxant may change from birth through childhood. Neonates and infants have a much higher volume of distribution and differences in redistribution, elimination, and metabolic rates. These vary with age. Young children may appear more sensitive to non-depolarizers, and there's some age-related variability. The ED95 is lowest in neonates due to that immaturity, but it's higher in children 3 to 10 years old and increases further towards adult doses in children about 13 years and older. Okay, elderly patients, the onset, once again, delayed. Okay, in our obese patients, non-depolarizers should be dosed on ideal body weight rather than total body weight to avoid excessive dosing. Okay, it's a muscle relaxant, not a fat relaxant. Full reversal is very important in the obese population, and we'll talk about that more in our reversal lecture. Now, in pediatrics, our non-depolarizers have a faster onset but a prolonged duration in infants, okay, compared to older children. But children generally recover faster than adults, okay? Oftentimes, children in that school age range require larger doses of non-depolarizers, and this can be due to their neuromuscular physiology and their overall better metabolism. So children tend to require higher doses. Infants, okay, are more sensitive, faster onset, prolonged duration. Now, we've talked about our inhalational agents, okay? These all potentiate. Des, SIBO, iso, halothane, okay? Nitrous oxide, propofol, pretty much nothing. Opioids, they don't really increase that, potentiate it, okay? So, in addition to that, we have some antibiotics that we have to watch out for, especially our aminoglycosides, our clindamycin, our tetracycline. And these antibiotics can work by inhibiting acetylcholine release at the neuromuscular junction, and this just augments that. You have more acetylcholine. We talked about the hypothermia, we talked about the magnesium, we talked about our local anesthetics, our quinidine. We talked about chronic anticonvulsant therapy, hyperparathyroidism, okay? So remember, keep your patients warm, okay? Make sure that if you're using magnesium, if you're using desflurane, make sure you take into account all of these things. If they're on chronic anticonvulsant therapy, okay, you're going to need larger doses. You're going to need to dose more frequently. Now, what are some adverse reactions? So there are some varying adverse reactions, okay, autonomic disturbances, histamine release, anaphylactic reactions, but these are kind of specific to each drug, okay? Autonomic side effects occur when they interact with the nicotinic and muscarinic cholinergic receptors. And pancuronium, for example, has those vagolytic effects, that increase in heart rate. Great if you're doing a nice big opioid induction, okay? Not so great if you have, say, aortic stenosis. Histamine release is another concern, especially with atracurium and mivacurium. You get that vasodilation, that hypotension, that flushing, that bronchoconstriction, okay? Not a great thing to get, okay? Allergic reactions, anaphylaxis, is rare but can be life-threatening. Rocuronium being the most common, implicated in all anaphylaxis of our neuromuscular blockers. Pay very close attention. This can happen. Be very, very cautious. Treat it quickly. Now, our autonomic effects of our neuromuscular blockers. They can interact with our autonomic receptors, including our nicotinic and muscarinic cholinergic receptors in the sympathetic and parasympathetic nervous system, as well as the neuromuscular junction. So these interactions can produce significant autonomic effects, changes in blood pressure, heart rate, systemic responses, okay? So, for example, curare, okay? This can cause some ganglionic blockade. This can cause hypotension, reflex tachycardia, histamine release, flushing and bronchospasm. Pancuronium has those vagolytic effects, okay? This action can suppress the modulation of catecholamine release, leads to excess sympathetic activity, increased heart rate, okay? You have to be very attention to this. You have to pay close attention to this. So the histamine release. Histamine release is a known side effect of certain non-depolarizers, especially those like atracurium and mivacurium. Okay, vasodilation, hypotension, tachycardia, bronchospasm. These are typically short-lived, okay, lasting 1 to 5 minutes, but they can be profound because you have to keep in mind though that you have multiple histamine receptors, H1 and H2. Be very cautious. Okay, asthma, COPD patients, no atracurium, no mivacurium. Careful with that bronchoconstriction. Watch out for that. To mitigate histamine release, they recommend giving these drugs slow, pre-treatment, but you know what? It's easier to stay out of trouble than it is to get out of trouble. Just use your steroidals for anybody who would not do well with an increased amount of histamine. So really just avoid atracurium and mivacurium. So now, what are those histamine-mediated cardiovascular effects? If you go down that road, if you did try to use that atracurium and mivacurium, and you get that histamine release, well, histamine, our H1 receptors are going to be on our vascular smooth muscle. You're going to get vasodilation, a drop in your blood pressure. Now, our H2 receptors, okay, some can be found in the myocardium. This can cause little positive inotropic and chronotropic effects, increase the contraction of our heart and our heart rate. Okay, this can lead to systemic vasodilation, cardiovascular instability. Curare, okay, a non-depolarizer benzyloxyquinoline. This can cause ganglionic blockade and leads to peripheral vasodilation and hypotension. Don't use it. Just don't do it. Now let's talk a little bit about our allergic and anaphylactoid reactions. Now, our neuromuscular blockers are among the most common causes of allergic and anaphylactoid reactions in anesthesia. Anaphylactic reactions are immune-mediated and revolve around the release of our IgE antibodies, and those bind to our mast cells, and that leads to the release of histamine and other inflammatory mediators. Anaphylactoid, on the other hand, are non-immune mediated, okay, but cause kind of similar symptoms due to the direct action on mast cells, okay? The incidence of these reactions vary depending on the agent used. ROC and SUX are typically the most frequently indicated of our neuromuscular blockers. So what are the clinical signs of anaphylaxis? Severe hypotension, severe bronchospasm, rash. Can lead to cardiovascular collapse. Immediately, 100% oxygen, epinephrine, fluids, antihistamines, corticosteroids. Preventing these reactions requires careful patient history, drug allergies, avoid triggering agents if possible. Skin testing can help you identify patients at risk, although it's not always reliable. So with all that being said, ROC and SUX are the most common triggers of our neuromuscular blockade and anaphylactic reactions. Be very careful. Now, of our steroidal muscular blockers, ROC, BEC, and pancuronium, they're not associated with much histamine release, okay? They can still cause the anaphylactic reactions, but it's rare. Rocuronium was implicated about 43% of the time in one study, and this is why it's very important to watch out. Now, cross-reactivity between our neuromuscular blockers, say you have somebody who has, they had rocuronium and they just had a terrible time, maybe just avoid all your steroidal, okay? But was it the antibiotic? Did you do a skin test? Did you get IgE testing? So if you have somebody who had severe cardiovascular collapse and had a steroidal before, maybe use Nimbex or cisatracurium, or do you need muscle relaxant? Can you get away with remifentanil? Okay, remifentanil will really, that's what we use in neurosurgery, and that's what we use in spine surgery to prevent somebody from moving and still do nerve monitoring. Okay, so there are other options out there. You don't just have to use another muscle relaxant. You can avoid muscle relaxants too. Now, if you get that histamine release, okay, especially that atracurium, especially that mivacurium, you're going to see that increase in your airway resistance, bronchospasm, okay? Be very careful in someone with asthma or COPD. If you have a hyperreactive airway, don't use a histamine-releasing agent, cisatracurium, or your steroidals. To prevent these complications, just don't use it. But if for some reason you do, okay, give them slowly, treat rapidly. Magnesium is great for a bronchospasm, but this is a histamine-releasing issue, so some epinephrine pretty quickly. Now, treatment of those anaphylactic reactions. Okay, recognize what's going on. Is your tube in place? Yes. All right, verify. Then work from there. Airway compromise can happen. Be very cautious. Epinephrine works by reversing a bronchospasm. It increases our blood pressure and reduces the severity of our other symptoms. Okay, fluid resuscitation. Okay, they're vasodilated. Give them some volume. Restore that perfusion pressure. Okay, you may need vasopressor support like phenylephrine or norepinephrine, antihistamines and corticosteroids. These are commonly used in treatment protocols for those with anaphylactic reactions. So epinephrine, quickest, 100% oxygen, epi, 10 to 20 mic, bolus pretty quickly. Okay, but if you've got cardiovascular collapse, give 100 mics epi. Get it in there, go from there, and then maybe do an epi drip. A lazy way to do it is if you have a liter of fluids, get a brand new liter, put 1 milligram of epi in there, shake it up. It's 1 mic per ml. Run it wide open. If you're having a true anaphylactic reaction, wide open until you get the effect you want. It's one easy way to do it. Poor man's epi drip. Now, prevention. Screen your patients. Ask questions. Look at old records. Maybe use other agents with a lower risk of histamine release. Cisatracurium. Do you need a muscle relaxant? You can intubate without a muscle relaxant. You can intubate with lidocaine. Okay, you can spray the vocal cords. You can intubate with remifentanil. You can intubate with propofol. Do you need a muscle relaxant if someone's had a terrible adverse effect? Can you use a muscle relaxant from another family? Did they have a bad reaction to rock? Maybe use Nimbex. Did they have bad reaction to Nimbex or atracurium? Maybe use a steroidal. Keep that in mind. Pre-treat as well. Okay, say you decide, hey, they had some hypotension last time, and it was post-induction. You can see it real, that's why I love electronic charting. I'll get on a tangent on this, but you can't hide a bad anesthetic with a good electronic record. It's going to show, it's going to bring everything over. So say they got hypotensive after induction. What can you do? All right, well, they're hypotensive after induction. You saw that. What did they give? Do you want to avoid some of those drugs? Do you want to avoid rocuronium? Do you want to avoid atracurium? Okay? Pre-treat as well. Mitigate those risks. Reduce risks. It's so much easier to stay out of trouble than it is to get out of trouble. So, the most common cause of intraoperative anaphylaxis, neuromuscular blockers. Okay? Antibiotics are implicated pretty highly as well. All right? This study says 15% antibiotics. But go with neuromuscular blockers, and then rocuronium for the most, and then sucks. How do you treat it? Like we talked about. You want to intubate. You want to secure that airway if this is true anaphylaxis. True allergy, you can have cardiovascular collapse. So there are some emerging trends. There are some newer agents being looked at. Ganticurium is one, and this would be rapid degradation. Basically, this is going to be a succinylcholine replacement. Okay, that's what's going to be next. We're going to get an agent that's going to be metabolized pretty quickly through probably non-specific esterases or a type of hydrolysis, and we're not going to use SUX. But until that time, SUX is going to be our rescue drug for laryngospasm. We want to have more selective relaxant binding agents like Sugammadex. Reversing these drugs is best rather than competitive antagonism. Because we didn't reverse before. Neostigmine, that's not a true reversal. Okay, guys, we have really dived into this. I've got a few more slides, but non-depolarizers, depolarizers, all of our neuromuscular blockers, we've talked all about this. It's vital that you understand this. This is such an important lecture, okay? I hope you've listened to every bit of it. I hope you've paused. I hope you've taken notes. Make the most of this. This is going to be your foundation that you're going to build. Okay? These drugs induce paralysis. They enable us to intubate, ventilate, and prevent the patient from moving. They act by interfering with neuromuscular transmission, either depolarizing the muscle or competitively inhibiting acetylcholine receptors at the neuromuscular junction. This leads to an inhibition of muscle contraction. Understanding the physiology is crucial for administering these drugs safely. The release of acetylcholine from nerve terminals at the neuromuscular junction initiates that muscle contraction by binding to our nicotinic receptors on the muscle fiber. Neuromuscular blockers disrupt the process, and their pharmacologic characteristics like onset, duration of action, metabolism determine their suitability for different surgical situations. Tailoring your neuromuscular blocker use to each patient ensures that you get effective relaxation and minimize your side effects. Okay, some statements to know. Neuromuscular blockers block skeletal muscle only, not smooth or cardiac. A motor unit consists of a single motor neuron and all muscle fibers it innervates. The postsynaptic membrane at the neuromuscular junction contains our nicotinic acetylcholine receptors. Paralysis begins with small, rapidly moving muscles, followed by those responsible for limb movement, and finally the diaphragm. You recover in the opposite order. Neuromuscular blockers act on those nicotinic acetylcholine receptors. Non-depolarizing neuromuscular blockers function by competing with acetylcholine for binding at that nicotinic acetylcholine receptor at our neuromuscular junction. They prevent depolarization, and subsequent contraction. Unlike acetylcholine, when they bind to that nicotinic acetylcholine receptor, it doesn't activate it. Non-depolarizers, they only need to bind to one alpha subunit, blocking that ion channel and inhibiting muscle contraction, This competitive inhibition allows for a controlled muscle relaxation. You have two main groups, your steroidals, and your benzoyl-isoquinolines. Pancuronium is our most potent and longest acting with those sympathomimetic effects. Vecuronium, less potent than pancuronium, is hepatically dependent. And rocuronium, about 1/6 as potent as Vec, quicker onset, great for an RSI. Now, succinylcholine. It's our only commercially available depolarizing neuromuscular blocker. It works by mimicking acetylcholine and binding to our nicotinic acetylcholine receptors at the neuromuscular junction. Succinylcholine, that molecule is actually two acetylcholine molecules bound together, and it has to bind to both alpha subunits. This causes continuous depolarization of the muscle membrane, prevents repolarization, and leads to sustained muscle paralysis. And this paralysis lasts as long as that membrane remains depolarized, preventing further contractions. Succinylcholine undergoes rapid metabolism through ester hydrolysis by pseudocholinesterase, and that's that enzyme that's going to break down the drug, It's going to break it down to succinylmonocholine and choline. However, you can have pseudocholinesterase deficiency, and this can lead to a prolonged neuromuscular blockade and lead to a slower clearance. In addition, succinylcholine can cause several side effects, including myalgias, changes in heart rate, bradycardia, tachycardia, elevated intraocular pressure, elevated intracranial pressure, hyperkalemia. It's an MH trigger. It's a dirty drug. I highly recommend avoiding it at all costs. Here is the chart from Nagelhout for our dose, onset, duration. Know this. Can't say it enough. Know it. And here are our agents along with the elimination mechanisms and a little bit more information. Guyton, Nagelhout, Miller, Stolting. The most important is Stolting and then Nagelhout. If you read those two chapters in Stolting and then that one chapter in Nagelhout, that's going to give you the best foundation. If you want more on the anatomy and physiology, if you want to look at Miller's, if you want to look at Barish, it's all there for you, but Stolting and Nagelhout are where you should start. Thank you very much. If you have any questions, please let me know.